in the same way it will go.
That's very good to hear.
Students will be back.
Is some vaccinations being done now in?
It's not to that scale, but still it is going on.
Okay.
It started on 16th. I think now it is, today is the seventh day.
Okay.
Jitendra, how many hosts you have made? Co-host or host?
Host is only one. Co-host, I think most of the organizing
All
committee.
organizing committee is the co-host.
Yes.
Okay, okay.
So I just made Jitendra as a host, so he will be taking care of all these things. Jitendra, can you make me co-host?
Unmute yourself, Jitendra.
Thank you.
Please unmute yourself also, Jitendra.
Yes, yes, sir.
And keep one thing vigilant on one thing particularly.
Yeah.
Sometimes background voices starts. So
Okay.
keep everyone muted during the presentation.
Okay.
Everyone muted except the speaker.
Okay, sir.
Dr. Sethi,
Ji?
just check whether Scott has joined or not.
No, sir. I have put an email also, but around 3-4 minutes ago.
Dr. Banu Singh is joining.
Okay, let him know.
Yeah.
Yes.
Yeah.
Good morning, Dr. Banu Singh.
Sir, you'll have to make him co-host.
Already made.
I made it.
Okay, sir.
Good evening, everyone. How are you?
Hello.
Hello, good morning.
Hello, Dr. Bhanu. Welcome to Gadwasu
Hello.
and webinar.
Hi, everyone.
Hello, sir.
Hello.
Number is increasing now very fast.
Yeah, we have
Yeah.
Playing it now.
Yeah, we get.
Because it is little bit early in India to start any program like this.
Yes.
9 o'clock.
Normally people go with the 10 or 9:30 somewhere. Because of our all experts from different parts, we started little early in this webinar.
Dr. Malik, that's almost like a half day for me because normally
Yes.
I start at 7 o'clock.
What is the time at your place, California?
Now it's 7 p.m. By the time I guess we finish, it will be more than midnight.
Okay.
I will try to, stay as much as I can.
Tomorrow is then holiday for you.
Of course.
Yeah.
It's a nice program. I won't trade that. I would like to listen as much as I can.
That's good. Thank you very much for being with us. Thank you. Do you have his number, Scott? We can call him on WhatsApp or some way.
No, I don't have his number.
Dr. Baljit, if he is having something, we can contact him. how to then go ahead. It's 8:35 now, Dr. Sethi.
Yeah, I'm trying. Dr. Baljeet, can you do you have any contact detail of Scott?
Sir, you can unmute yourself, sir.
I don't have Scott Napper's telephone number, but I'm sending a text message to Volker Gertz, who's the director of the video, to see if he can quickly contact Scott and let me know if we can reach him.
Okay, thank you. Welcome, Dr. Yadav. Professor Yadav has joined.
Dr. Jitendra, can you share with me the quick link to join so that I can send Scott
Okay, sir, I'm sharing, sir.
email?
Sir, I have shared to your email, sir.
Thank you. Jason.
Dr. Sethi,
Hmm?
is there any update?
No, sir.
Or otherwise, what we can do, we can plan with the second one. And until he will finish, we will be in contact with Scott Nipper.
Okay.
Because it's not, because already we are 10 minutes over. Dr. Baljit is saying something. Please unmute him. Jitendra. Dr. Baljit is waving hand.
Yes, yes.
Please, yes.
So I spoke to Volker Gertz, the director at Vido. It's interesting, even he doesn't have his cell phone number, and he is looking it for it. I just sent Scott an email, and because Volker tells me that he reads his email all the time, so I'm not sure whether Sethi has sent him an email yet or not.
I have sent him email as to requested him to log in, and then I have sent the link also, but there is no response.
Again, he is muted, Jitendra.
Sir, you,
Okay, what I will do is that I'm going to mute myself for a moment, and I will try somebody else to see if they have a cell phone number for Scott, and give me a few minutes.
Dr. Malik, should we start the proceedings?
I think we can proceed with Dr. Shovik. Already he has joined.
Just give me 1 minute.
Okay, Dr. Baljit.
Well, anyway, so now there are two people scrambling to find Scott's telephone number.
Okay.
Make sure as a vice president in 10 days time, I will put it a negative point in his file. So either you can begin with the next speaker or you can wait for another 5 minutes, then we will get some answer. It's up to you. I will continue with my effort here.
Yes, Dr. Sethi, what we can do, let's start with the proceedings because
Yes.
everyone is waiting and more than I think 160 now delegates are with us.
yes, sir.
So better to start with it. And if within 5-10 minutes he joins, we can start with his inaugural lecture. Otherwise, we will invite Dr. Shobik. Already he is with us and he will start with his lecture.
Sure.
Excellent plan. Thank you very much. I will keep on following up to see if I can find him.
Thank you very much.
Thank you very much, sir. So, welcome everyone, and this is a bit late start. The reason being, there are some technical issues. Sometimes the time differences with the um, uh, our guest speaker being located overseas at a far distance place, so the time difference. So my apologies for that, and welcome to this international webinar on COVID-19 pandemic. It is being organized by the College of Animal Biotechnology, Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana, in collaboration with Indian Virological Society. I request Dr. Malik for the welcome address.
Thank you very much, Dr. Sethi, the organizing secretary of this webinar that is jointly organized by College of Animal Biotechnology, Guru Angad Dev Veterinary Animal Sciences University at Ludhiana, Punjab, and Indian Virological Society. That is a well-known society, not only in India, but abroad also. And I'm lucky to have some of our legendary people from that Indian Virological Society who have joined us. Professor Yadav, Professor Uppal, Dr. R.K. Singh, who is the current president of Indian Virological Society. And I am the General Secretary of Indian Virological Society. So I take this opportunity to just brief about the society and the college that is holding this important topic. And I can see the participation that within a couple of days that we announced this webinar, and within couple of days we crossed the 500 mark of the participation. We have a limit to accommodate this number. That's why we restricted, not allowing the rest of the participants. And this is a big apology from our side. But we are hosting this as a in the live stream mode on the Facebook of Gadwasu, our college, and our society, Indian Virological Society. So people can be benefited by joining from that area, from that Facebook or YouTube also. So a little bit I will brief about the society. This society is a 36 years old society, well established, and it is a member of International Union of Microbiological Society. That is very important thing for the society to have this honor. And we are also part of the World Society for Virology, which is spread across nearly 80 countries with 2,000 virologists across the globe. So our Indian Virological Society component is a member of 2 international bodies. And the unique thing about Indian Virological Society is that we are having people from different disciplines. Sometimes we have very small societies focusing particular area, but in this society, we have plant virologists, we have medical virologists, we have aquatic virologists, and the veterinary. So this is something uniqueness of this society, that nearly 500 active members from different disciplines in India and across world are the member of this virology society. The society is running one journal that is published by the Springer, and since its inception in 1984, it is continuously publishing this journal. Earlier, the name of the journal was Indian Journal of Virology, and then it was renamed in 2014 as Virus Disease. And just last year census, I will tell, we got the papers from 32 countries to be published in this journal. This is a very good journal, and it has been indexed in many important indexing agencies. So I request you all to visit the IVS website and go through the number of awards people from this society. They are taking part in number of international engagements, and this society also publishes a newsletter on the new virus research, something which is pertinent to the public, and it must reach to them in a very brief manner, in a very normal language. So that also we publishes regularly from this society as a newsletter. So regarding our College of Animal Biotechnology, and just to let you know that because people are from different parts of the country, from different disciplines, not only virologists, even some of our media persons have also joined, and the persons from basic background also, they have joined this because the concern of today is what Dr. Baljit was also telling, pandemic. We have to discuss, we have to coin something new for the coming days so that such threats are not a problem for the future. So we must be prepared for the future. So number of people are there. So let me brief that this college is earlier it was School of Animal Biotechnology. Then in 2017, we established it as a College of Animal Biotechnology. And we are offering BTech, Biotech, MTech, PhD programs from this college. And this college is a part of the Guru Angad Dev Veterinary and Animal Sciences University. And just for the notice of everyone, this is the ranking 1 university of the country in veterinary sciences. So we are proud of everyone who has contributed for making this university to this level. So this is all about the college and IVS, and the topic which has been coined for this webinar. I compliment the organizing committee, particularly my colleague Dr. Sethi, who is the organizing secretary, joint and the co-organizing secretaries, Dr. Ratan, Dr. Gowda, Dr. Jitender. Everyone has worked very hard, and they planned in such a way and came out with a proposal that we will organize one webinar on COVID pandemic and the concerns and perspective, because it was a big problem when it started in December 2019, and within 1 year so many changes occurred. It revolutionized the biotechnological approaches and number of approaches for diagnosis and vaccine. So many advancements were done, and sometimes people are aware of it and sometimes they are not. So what we planned, that why not to have an interactive session, why not to plan one webinar with international experts, national experts, and our very senior colleagues from the Virological Society to discuss over this issue of pandemic. This pandemic has crippled our lives, and because I am from veterinary, I must comment that even our animal husbandry suffered a lot. So this has not only affected the public health, but this affected the animal health also. So this is a zoonotic concern, and that's why we included many of our colleagues. And I'm thankful to the experts from international fraternity, Dr. Scott from Canada, and thanks to Dr. Baljit for being with us on this occasion. And he is one of the mentor for this university, our college. And we are thankful to him for joining us on this. Definitely, these are some odd hours in Canada and US. My friends are from St. Kitts, that is West Indies Caribbean part. Dr. Souvik Ghosh, who is the director of zoonosis at the University of St. Kitts. Then Dr. Bhanu, he is a senior director at Gilead, US, California. He also consented to be part of this webinar. And my friend from UAE, and he is working at Uppsala, Sweden. He will be with us in One Health concept. He will just speak on that issue. Then some of the national experts are from Integrated Disease Surveillance Program of National Communicable Disease Center of India. Then the manufacturer of the vaccine from BioVet, the Vice President, Dr. Panduranga will be with us. Dean Medical College KGMC from Lucknow. He is with us, Dr. Shailendra, and he is the also editor-in-chief of the Virus Disease Journal. And he has recently published a special issue on the coronavirus also. And a very good book from the Springer also he published recently. So he will be with us along with some of the panelists, senior virologists from the country. We have requested them to be in the evening hours with us so that we can discuss on some issues and some strategies that we can make the roadmap for the future to tackle some of the problems. One name I forgot, I apologize, that Dr. Laura, she is from Australia. She is also speaking on, she is from biosecurity section of animal health from Australia. So she will be with us for one of the lecture. So without taking much of the time, I just request the organizing secretary to please proceed.
Thank you very much, Dr. Malik. And let me say that it could have been not possible without the help of Dr. Malik. He is so kind. He helped us a lot, planned everything, even minute details. I could see his messages even in the late night that change this flyer. I need a minute changes. Thank you very much, sir, for putting so much efforts. And same thing with Dr. Baljeet Singh. And even yesterday night, he was, curious about how you are planning this seminar. Thank you very much, sir. So there is some change. We will go ahead with Dr. Shobhik Ghosh. And Dr. Shobhik, he's a professor, infectious diseases, Department of Biomedical Sciences, as well as he's DVM course director for virological section, Director, One Health Center for Zoonosis and Tropical Veterinary Medicine, Ross University School of Veterinary Medicine, West Indies. He has an interdisciplinary teaching and research experience in the fields of veterinary and medical virology, molecular epidemiology, viral genomics, zoonosis, and One Health. He is two-time recipient of the Care Award for his contribution to teaching. Today, he will be speaking on the understanding and combating COVID-19, a veterinarian's perspective. Dr. Ghosh, please.
Yeah, thank you so much. Let me share the screen. There it is. Before I get started, I have been teaching virtually for most of the past year because of this COVID-19 situation. And this is something I always ask my class before I get started. So I'd like to ask all of you, can you hear me properly?
Yes, sir, go ahead.
Can you show by a thumbs up or something like that if you can hear me properly?
Yes, you
Excellent.
are audible. Yeah.
And can you see the full screen? Can you see the slide full screen?
Yes.
It's visible,
Excellent.
sir.
Thank you so much. Yeah, pardon?
Yes, yes.
Yes, sir, go ahead.
Please go ahead.
Okay. Okay, thank you so much. Yeah, so good morning to all in India and good evening to colleagues in US, Canada. So I would first like to thank the organizing committee, Guru Angad Dev Veterinary Animal Science University, and the Indian Virological Society for this opportunity to speak in front of such an esteemed gathering. And I'd like to wish again greetings from West Indies to all my esteemed colleagues in India and other parts of the world, and to my dear students. So today I'll be speaking on understanding and combating COVID-19 from a veterinarian's perspective. The reason for this talk is just before this meeting, I was checking PubMed. And when I typed in COVID, I found there were 97,000 papers in the PubMed. And then when I typed SARS-CoV-2, I found that there are more than 55,000 papers in PubMed. Now, if you look in all those papers carefully, I know that it is going to take a lot of time, but if you look into all those papers carefully, you will see that most of these publications are on these coronaviruses from the human perspective. Of course, there are some papers that are on coronaviruses in animals, but these are primarily from the zoonotic point of view. That is, how COVID-19 or SARS-CoV-2 got transmitted from animals to humans. Unfortunately, there are few papers that literally have compared animal coronaviruses with human coronaviruses. We developed interest in coronaviruses in humans after the SARS outbreak in 2000 to 2003, which luckily was very limited, nothing compared to what's happening now. But animal coronaviruses have been studied for more than 3 decades. And there is a wealth of data available on animal coronaviruses. And when you sit down and you start comparing those with the information that is available on COVID-19, you will be surprised to know that there are some striking resemblances. There are some similarities. And this information can be extremely crucial in our understanding and combating COVID-19. So that is the basis of my topic today. I expected that the speaker before me will do the honor of introducing COVID-19, but since you know I am the first speaker, so these are some of the few details about this virus. This is a RNA virus. Of course, it belongs to the family Coronaviridae. As pointed out, the first official case was reported in China in December 2019, though it is believed that the virus may have been circulating way before that. And of course, based on genome analysis data, it is proposed, and there is strong evidence that the virus may have originated from bat and may have been introduced to humans through an intermediate host. The fingers point towards pangolins now. So there may or may not be an intermediate host involved in this transmission process. And of course, the virus is considered as a respiratory virus because of its clinical presentation. But as more and more clinical evidence is accumulating, it is now known that the virus has impacts beyond the respiratory system of the human body. And of course, what is the impact? And because of the impact, all of us has assembled here together to discuss COVID-19. As of January 22, 2021, more than 96.2 million people are infected, and the numbers may have increased because these are the numbers in the morning, and the numbers have significantly changed now that it is midnight here. More than 2 million people have died. 219 countries have been affected. More than 11 trillion US dollars have been lost. And of course, there are other impacts on health, impacts on human welfare, and impacts on society. So, of course, this is a really, really catastrophic situation. Long time back, when I was a kid, my grandparents used to tell me that if you are united, we will stand, and if you are divided, we will fall. Everything will fail. Imagine the audacity of a virus that has changed everything. For the first time in the history of mankind, divided we stand and united we fall, right? All these social distancing measures, we should not come in close contact with each other to survive, right? So imagine a virus that has the audacity to challenge the basic tenacity of survival of mankind, right? So this is what this virus is doing now to mankind, which is really a very serious problem. However, how can we combat COVID-19? We can combat COVID-19 if we really believe in the concept of united we stand, divided we fall. And this is only possible if veterinarians, medical doctors, environmental specialists, they join hand together to understand and combat this pandemic. And that is where, this talk is based upon, where I shall be speaking from the veterinarian's perspective. How do I look at COVID-19 from the veterinarian's perspective? The role of veterinarians is of paramount importance. Of course, we play a very important role in ensuring the health of livestock, the health of companion animals. We play a very crucial role in food safety and security. but at the same time, a significant number of pathogens that are being reported in humans are derived from animals. As you can see, some of the statistical data here obtained from the OIE website, more than 60% of the human diseases which are of infectious nature may be derived from animals. So veterinarians have to play a very important role in combating zoonoses such as COVID-19. And why do we need veterinarians to look at COVID-19? Because without veterinarians, you do not see the complete picture. As you can see here, the camera is actually showing something, but actually something else is happening. So veterinarians also play a very important role in showing the full picture so far as zoonotic pathogens such as coronaviruses are taken into consideration. So the first and foremost important role is tracing the origin of the zoonotic coronavirus. You know, zoonosis is a very complex puzzle. The virus is transmitted from animal to human. There may be intermediate animal host involved. And unless and until you have all the pieces of this complex puzzle, you do not know how the virus has been transmitted from animals to humans. So it is very important to connect all these different components of a puzzle to get the proper picture, the complete picture. And that is where veterinarians play a very important role alongside medical professionals. Before I go into the details, I'll just like to define a term that is recombination. I know that most of my colleagues are esteemed virologists, but there may be some students among the audience. And this is what recombination means. Like you have two viruses infecting a cell, and then you have a new progeny virus where a part of the genome is from one parent virus and another part of the genome is from another parent virus. And this recombination plays a very crucial role in coronaviruses. So until now, based on published literature, there has been 7 instances where coronaviruses have jumped the species barrier from animal to humans. Of course, all of us are aware of the SARS outbreak that occurred in 2000 to 2003. Around 800 people died. It spread to some parts of the world, but it did not expand. But unfortunately, things changed a lot with this COVID-19 or SARS-CoV-2. So in 2005, although the SARS virus that caused the SARS outbreak that originated again in China and then spread to other parts of the world was reported in 2000 to 2003, the origin of SARS, the search for the origin of the SARS virus gained momentum only in 2005. And during the surveillance of bats, they found viruses that were very closely related to the SARS virus based on genome sequence analysis. And then they found similar viruses in the civets. And based on the genome analysis, epidemiological data, it was proposed that the SARS virus underwent recombination events in bats, and then eventually it got transmitted from bat to civets. These civets were exposed to stress in the wet markets, where the virus underwent further mutation, and eventually they got transmitted in humans. But studies in human population around the bat caves in China has revealed that humans staying near bat caves in China have high antibody titers against the SARS virus. So this raises another aspect, that was it a direct transmission from bat to humans? And this is something which requires conclusive data, because what evidence is available is still serological. And then came the next big one, that is the MERS, a Middle East Respiratory Syndrome. Now, the history of MERS is very old. It is believed, again, based on genome analysis, which is very crucial, that the MERS virus was introduced from bats to camels almost 30 years back. And then the virus underwent mutations, recombinations, and eventually it got transmitted to humans. And the first recorded transmission was in 2012. MERS was not associated with large-scale outbreak other than 2 outbreaks. There were many sporadic cases. And then came the big thing, the SARS-CoV-2, which is known as COVID-19 more popularly. Now, when they characterized the SARS-CoV-2 virus, they found that 96.2% of the genome sequence was closely related to that of a bat coronavirus, which was reported from a sample collected from a bat cave in 2013. So there was a constant surveillance study going on with bats, and they had this database. And when this COVID-19 genome was sequenced, they found that it shows a 96.2% match with a bat coronavirus from a sample collected in 2013. surprisingly, the critical receptor binding domain, which is very crucial for the virus to attach to the host cell, one of the first steps of the virus to attach before it infects the host cell, was different from this bat coronavirus. It shared only 85% sequence identity, and 5 of the 6 crucial amino acids that are required for this binding were different. Again, they found these coronaviruses in pangolin, where surprisingly again, the receptor binding domains encoding sequence of pangolin coronavirus was more than 97% identical to SARS-CoV-2. And based on these findings, it has been hypothesized that it may be possible that SARS-CoV-2 was transmitted from bats to pangolins, and eventually it got transmitted from pangolins to humans. And this may not be surprising because pangolins are being sold in the wet markets in China, where wildlife and humans come in close proximity to each other. But there are interesting twists to this hypothesis. It is also believed that it may be possible that the virus was introduced into the humans long time back. The virus underwent mutations or cryptic spread in humans, and eventually it evolved in a highly pathogenic form, which eventually resulted in a pandemic strain. And then, the fact that that with the SARS virus, antibodies have been reported in humans living near the bat caves. Detection of different genetic components of the SARS coronavirus in bat caves from different bats, which if you take together may constitute a SARS virus, led to the hypothesis that it may be also possible that the virus was directly transmitted from bat to humans without an intermediate animal host. So still there is no conclusive findings on the exact route of transmission of the SARS-CoV-2 from animals to humans, but definitely it is a bat virus that got transmitted to humans with or without the involvement of an intermediate host. And it is surprising, this virus has adapted itself so well in humans. It is spreading so rapidly in humans. It is basically present all over the world, right? But for a veterinarian, this may not be that surprising, because if you look into the huge pile of literature that is available on animal coronaviruses, there has been similar instances. One of the best examples in this regard would be the transmissible gastroenteritis virus of pigs. This virus, based on genome study, it has been hypothesized that it originated from canine coronavirus. Similarly, feline coronavirus 2 is believed to have originated from recombination events between feline coronavirus 1 and canine coronavirus 2. There has been instances where, coronaviruses have been transmitted from bats to pigs. And one of the best examples is porcine epidemic diarrhea virus. Based on genome analysis, it is hypothesized that the ancestor of PEDV may be pigs. More recently, in 2016, the swine alpha enteric coronavirus, which caused outbreaks in China, was traced to bat. And there was conclusive evidence that this virus was transmitted directly from bats to pigs, which was a very big warning signal that bat coronaviruses can be transmitted to humans too. But nobody realized that, right? And if you look very carefully into the evolution of coronaviruses, you will find that based on genome analysis, it has been hypothesized that the ancestor of the alpha and beta coronaviruses are bats, whereas the ancestors of the gamma and delta coronaviruses are birds. And rodents and cattle may have played some role in the evolutionary process of the embecoviruses. So if you look into the evolution of coronaviruses, you will find that transmission of the virus between heterologous host species and adaptation of the virus to the new host species, there is literature available with regards to animal coronaviruses. So this is something that is not new, right? And that is why, studies on animal coronaviruses is very important. The next thing was COVID-19 is a very enigmatic virus. It is very mysterious. It has taken all the researchers, all the virologists in a wild goose chase, right? However, again, if you look into the data available on animal coronaviruses, you will realize that it is not only COVID-19, but animal coronaviruses are also enigmatic, which means that the COVID-19 or the virus of humans can be enigmatic because that is the nature. Initially, coronavirus was thought as a respiratory virus. Definitely, most of the significant clinical presentation was that of respiratory. But then, other involvement of other organs started being reported. And now there is also evidence that the virus is shed in feces, and sometimes it is shed in feces for longer duration. Then it is being detected from a nasal swab. So this virus is very mysterious. There is no conclusive data on the exact pathology of the virus in the human host. Now, if we look into animal coronaviruses, let me show you some instances. There are lots, but some of the very, very important instances where coronaviruses have showed strange behavior. Enteric canine coronavirus was a mild enteric pathogen. It used to cause mild diarrhea. Fine, self-limiting. However, certain changes happened in the virus, which we don't know. The virus became pantropic, resulting in a systemic disease. This is called pantropic canine coronavirus. Highly virulent, systemic, lesions in different organs across the body, high mortality rate. Nobody knows until now what exactly were the changes in the virus genome that transformed a mild enteric canine coronavirus into a systemic, highly virulent pantropic coronavirus. Another very good example is that of transmissible gastroenteritis virus, which causes severe diarrhea, high mortality in piglets, and a deletion of 600 nucleotide at the 5' end of the S protein encoding gene. Voila, this enteric virus transforms into a mild respiratory coronavirus, causing mild respiratory disease in pigs. And it has been postulated that the respiratory coronavirus, which is mild, confers partial immunity, which has resulted in the reduction of mortality associated with transmissible gastroenteritis virus. So imagine deletion, and the virus transforms from an enteric virus to a mild respiratory virus. But one of the best examples of the enigmatic nature of the coronavirus, which has striking resemblances to COVID-19, is that of feline infectious peritonitis virus. Now, feline coronaviruses typically cause diarrhea, mild diarrhea in 70% of the cats, but then there is 1 to 3% of these unfortunate cats that develop a very systemic disease called feline infectious peritonitis. And this is a comparison of the pathogenesis of COVID-19 in humans and the pathogenesis of FIPV in cats. So what happens in cats? The hypothesis is that the feline enteric coronavirus accumulates mutations. The guess is that it is perhaps these mutations which are responsible for the transformation of the feline enteric coronavirus into the virulent FIPV. And this FIPV has an affinity or tropism to replicate efficiently in the monocyte or macrophages. And once this FIPV infects monocyte or macrophages, these monocyte or macrophages, they not only spread the virus across, but they release cytokines, chemokines, which causes a systemic infection. And then, there is production of antibodies. Now, these antibodies, instead of neutralizing the virus, they bind to the virus, and the virus utilizes the Fc receptor of the antibody to infect more monocyte and macrophages, resulting in a more severe infection. And what you see is the effusive form or the wet form of feline infectious peritonitis. So as you can see, this pathology is quite strikingly similar to what we see with the SARS-CoV-2 or the COVID-19 virus, where the virus causes pyroptosis. There is release of PAMs and DAMs. This attracts the inflammatory cells, monocyte, macrophages, T cells, release of chemokines. Then there is a formation of pro-inflammatory feedback loop. More cells are recruited. And then if there is a defective immune response, there is a cytokine storm, which is believed to be one of the causes of high mortality. There is no conclusive evidence that SARS-CoV-2 causes antibody-dependent enhancement, something that we'll be discussing later on. But there is evidence that SARS causes antibody-dependent enhancement, and the cytokine profiles seen in COVID-19 patients with poor prognosis are indicative of antibody-mediated monocyte or macrophage activation. Okay, now what is the most important thing at this point? Vaccine, right? We already have vaccines that are being administered, and we have so many vaccine candidates. But by the way, did anybody think, what is the status of animal coronavirus vaccines? Do they have some vaccines against animal coronavirus? Oh, I was not aware. Are they working? Let's see. Now, there are vaccines against animal coronaviruses, and based on published data, none of the vaccines afford complete protection. Although the vaccines do afford protection, studies have shown that none of these vaccines against porcine coronaviruses, avian coronaviruses, or the bovine coronaviruses afford complete protection. Another concern is the short duration of immunity. With animal coronaviruses, it has been shown that the vaccine confers a short duration of immunity, And with the IVV vaccine, it has been also seen that there may be shedding of the virus following vaccination. And the correlates of protection against coronaviruses with regards to the immune response is not properly defined, because this is very important with regards to the route of vaccination, intranasal or parenteral, because that will elicit different kinds of immune response, right? But with regards to COVID-19, this data is not very conclusive. But similarly with vaccines against animal coronaviruses also, this data is not very conclusive. And then there is another issue that is very important is strain diversity. And recently we are aware of variants, right? COVID-19 variants that have emerged in UK, COVID-19 variants that have emerged in South Africa, and they're skeptical that if these variants, makes these vaccines less efficacious. With animal coronaviruses, we have seen similar instances, right? With IVV, with PEDV, there has been, mutations or recombination resulting in emergence of strains which may render the vaccine less effective. So constant monitoring of the viral genome is very crucial. And then was another very important point of concern is antibody-dependent enhancement. So what is antibody-dependent enhancement? Typically, when you have antibodies, when you're infected with a virus, your immune system identifies the virus, there is production of antibody, as you can see in this very easy to understand cartoon, and the antibody tries to neutralize the virus. But with coronaviruses, at least with feline coronaviruses, it has been seen that there are production of sub-neutralizing antibodies which cannot eliminate the virus. Rather, the virus binds to the antibody, and then it uses the Fc receptor of the antibody to infect more monocytes and macrophages, which causes a very severe spread of the infection and very severe disease known as the wet form of FIPV. Now, no conclusive data is available with regards to antibody-dependent enhancement for COVID-19 or SARS-CoV-2, but studies with SARS coronavirus have shown that when lab animals are immunized with the SARS coronavirus, they may exhibit antibody-dependent enhancement. And as I said before, some of the cytokine profiles of SARS-CoV-2 patients with poor prognosis matched that of antibody-mediated monocyte or macrophage activation syndrome. So antibody-dependent enhancement is one of the very key aspects that needs to be investigated with regards to SARS-CoV-2 because this has implication on vaccines. So in case of feline coronavirus, studies have shown that immunized cats following natural infection, can develop more severe disease, and this has been attributed to antibody-dependent enhancement mechanism. Considering these and considering the preliminary evidence of ADE with the SARS virus, not the SARS-CoV-2, but the SARS-CoV-1 virus, it is very important to look into this aspect. Now, of course, one of the important aspect is contact potential. We have opened the Pandora's box. We have opened the Pandora's box. The virus is out. So what you reap, you sow. For we opened the Pandora's box. So one of the very crucial factors is that these kind of zoonotic pathogens will cause havoc if they come in contact with humans. So contact potential is one of the very key components of transmission of this kind of zoonotic pathogen. Climate change, deforestation, and illegal wildlife trade are some of the factors that facilitate this kind of zoonotic transmission. And of course, we know that history repeats itself, right? But we never learn. And if we don't learn, what is going to happen? Very soon we'll become history. That is what is going to happen, right? So we should learn from this kind of situation. So we should stop deforestation. We should fight against climate change. We should advocate against climate change. We should ban, wildlife trade. We should ban wet animal markets because these are the breeding grounds of these kind of zoonotic pathogens, and they can be easily transmitted to humans because of the close proximity of humans to wildlife. And then equally important is to perform constant surveillance study for zoonotic pathogens in wildlife. When the SARS outbreak occurred in 2000 to 2003, it was only during a surveillance study in 2005 they identified the virus, which was in bats, which was closely related to the SARS virus, leading to the conclusion that the SARS virus may have been derived from a bat coronavirus. We were more lucky this time because there has been a surveillance study that was going on in China in collaboration with the EcoHealth Alliance, And when they sequenced the COVID-19 or the SARS-CoV-2, they found a virus in their database that was collected in 2013 that showed a 96.2% match, which immediately led to the conclusion, well, this may have been derived from a bat. So surveillance for viruses in wildlife is of paramount importance. And then it is equally important to perform surveillance study in humans who are in close proximity to wildlife. When they did a serological study in humans staying near bat caves, they found high antibody titers against the SARS virus. This indicates that the virus may have been transmitted from bats to humans a long time back, and there may have been repeated transmission, but since there was no severe clinical sign, or since it did not spread that rapidly, nobody noticed it. But this was a warning sign, right? Similarly, it is equally important to perform surveillance studies in livestock living in close proximity to wildlife. One of the very important warning signals that bat coronaviruses can enter humans again and again was that of the swine alpha enteric coronavirus outbreak in China in 2016, which was actually transmitted from a bat to pigs. So these are some of the warning signs we can get if we perform surveillance studies in livestock in close proximity to wildlife. Coronaviruses has a quite large genome. It is around 30 kb, which means that the virus genome is more vulnerable to genetic changes. However, it has proofreading activity, but the virus replication rate is quite high, which may compensate the benefit that we may get because of the proofreading activity. And again, because of template switching ability, the virus is prone to recombination event. So it is very important to survey to study the genome of the coronavirus in animals and humans, because the genome is vulnerable, it is prone to changes, as already evident from the variant strains that we are seeing just in such a short period of time from South Africa and UK. And this is a recent report in Nature that mentions a concern about the COVID variant with regards to eluding immune responses and creating doubts on the vaccine efficacy. So what are the takeaways from this lecture? That first of all, coronaviruses can be very enigmatic. And how do we know that? We did not have to wait for COVID-19. The evidence that we had from animal coronaviruses provided the foundation or the platform that these viruses are very enigmatic. They can change their tropism, they can develop pantropism, and they can cause a wide array of clinical signs. So it is, and of course, the effect of the virus on the immune system, which can go against the body, such as antibody-dependent enhancement. So it is very important to have a thorough knowledge of animal coronaviruses, because this will help us to understand and combat COVID-19. So these are some of the key points from this lecture. And what is the conclusion? The conclusion is that we have to be one, to be united, and to fight this disease, we have to be one. So professionals from different areas of expertise, medical people, veterinarians, researchers, sociologists, financial people, bureaucrats, they should all assemble together. Different stakeholders should come together. And that is only when we can combat such a deadly pandemic. Because of time constraint, I could not elaborate on all aspects of comparison between animal coronaviruses with COVID-19. But recently, with my good colleague, Dr. Malik, we published a review in Microorganisms, where this is a comprehensive review where we have tried to draw comparisons between SARS-CoV-2 and the different animal coronaviruses. What I showcased in my lecture are some of the highlights from this review article. And that is the reason I come from the Ross University School of Veterinary Medicine in St. Kitts, West Indies, and our research mandate is One Health. Although we are a vet school, we amalgamate our research with animals and humans, and we always work in the overlap area from the One Health perspective to study zoonosis. So One Health is of paramount importance to combat diseases, pandemic situations like those caused by SARS-CoV-2 known as COVID-19. So thank you for your kind attention. And if you have any questions, I'd be happy to answer those.
Thank you, Dr. Shovik. Thank you very much for an elaborative talk, and which will help us to understand the basic facts related with the structure, transmission of COVID-19, as well as you put questions and role of veterinarians in combating zoonosis such as COVID-19 and challenges related to this pandemic. So Dr. Bedi, if you are ready, we will take 3 more questions for Dr. Shovik.
Sure, sir. I'm waiting in the chat box for some questions to be raised by participants.
Okay. So Dr.
Uh,
Shovik, I was interested how, how far these animal models are true for this COVID-19 research.
Uh, some some work definitely has been done on animal models, and as I said before, and this is not of course COVID-19 because COVID-19 is relatively new. But as I was saying, one of the very critical aspects that is a little bit scary, I don't want to sound very pessimistic, is that of antibody-dependent enhancement. Now, with the SARS virus, not this COVID-19, but the SARS virus that caused the outbreak in 2000 to 2003, they found that non-human primates were immunized, and when they were treated with the natural infection, they did show antibody-dependent enhancement. So these are some of the aspects which shows that animal models are really important.
Thank you.
But again, COVID-19 is relatively new, and there are labs that are trying to develop and have already developed animal models to study different aspects of the virus. But still now a significant chunk of the information remains inconclusive.
Thank you, sir. Dr. Bedi, are you ready with some
Thank you, sir.
questions? Yeah.
So still I'm waiting, but I'm thankful, Dr. Ghosh, you have given a very wonderful lecture.
Thank you.
I have a one query, sir. Since you have told that One Health approach is a very useful to control such type of pandemic, and as far as COVID-19 cases which appeared in animals, they have a link with the human cases.
That's right.
Yeah,
Yeah,
sir.
that is an yeah, I'm sorry I didn't allow you to complete your question.
No
If you
problem,
may,
sir.
please.
Sir, can there are some chances, or it can be explored that it may occur as a reverse zoonosis, that those animals which have been affected with
Yeah, that's a
those
very
animals?
interesting question. Yeah, that's a very interesting question. Well, all of us are aware of that mink incident that happened in Europe. It kind of eliminated the mink industry in Europe, right? So the virus got transmitted from humans to mink, and then there was a reverse zoonosis from minks to humans, which prompted some of the European countries to eliminate or to cull all the mink population. And this was a devastating loss to the mink farming industry. There has been instances where this virus has been transmitted from humans to cats, humans to dogs, humans to wildlife, and of course mink. And a paper was published in the New England Journal of Medicine where they did an experimental study where they showed that the virus can be transmitted from cat to cat. There was a report that was published by OIE where they highlighted these aspects, and it states that the virus can propagate well in ferrets and cats, whereas the virus does not survive well in dogs. So that is what the OIE report states.
Thank you, sir. Sir, we have one question.
Yeah, please.
That can we estimate the, sir, impact of COVID-19 pandemic on the economy of animal health sector?
That's a very interesting question, and as Dr. Malik was speaking about this during the introduction, and I felt that he had raised a very important point. Yeah, we are more concerned about humans, but unfortunately, I don't know what is the statistics with regards to the economic impact of COVID-19 on animals. But I guess as we get more acquainted with this catastrophic scale of this pandemic, this is an area that will definitely be looked into and has to be looked into because not only livestock, but pet animals are very important. Livestock health has implications on food safety and security, especially in developing countries, as well as for the livelihood of people in countries where agriculture is a very important component. And pet animals have a impact on the society, on the mental health of people. But unfortunately, not much is known as of now with regards to the economic impact in livestock or in pet. But this is a very important point that needs to be investigated.
Thank you, sir.
Dr. Souvik, can I add something to your answer here?
Please,
This
sir.
is Banu.
Sure,
Can you hear me?
Sure, sir, sure.
So this is a very good question or topic which you raised, the impact on the animal health, and I think it depends which way you are looking at. I can tell, we have gone through the period of lockdown where people were locked in their home or isolated in their home. And I was reading some reports where all of a sudden the demand for pet, because people were looking, we have to understand a lot of people live by themselves, and the demand for companion animals was very high in that time. So it's in a way it was very useful for us, but at the same time, because animals were not allowed to go outside, I don't know what will be the impact. Like if you have your dog and you're not allowing them to go for months outside the apartment or have limited activity. So I think only time will tell. There are different aspects on the health of our companion animals.
And sir, that was a very important point. And if I add, with your permission, that at least I can speak from the US perspective, because this got reported in the news. Many of the farmers were forced to discard tons of milk or cull a lot of pigs because of this lockdown. The supply chain has totally broken down. So at least, based on the news that I read of what's happening in the US, it did have a significant impact on farmers. But what was the extent of economic damage? I shouldn't comment unknowingly, but I feel that as of now, that statistical data is not clearly available.
I think there was one incident in Purdue Farm, which is a major poultry chicken industry. Any supermarket you go in the US, you will find the Purdue chicken, and that originates from one of the big farm in somewhere in Wisconsin, and they have about 5 or 10 workers which were tested positive. Obviously, these days the news travel very much fast, and the people were so fearful that they stopped discarding the chicken, and the supermarket start throwing all that meat. Although, who knows what the chicken was, were even a source for transmission or not. But a lot of impact, I would say, on the animal health in general.
That's right, sir.
So I do have a question for you, Dr. Souvik. I think you emphasize quite a lot on the antibody-dependent enhancement. I would say, and I'm a veterinary pathologist, so I think I've seen a lot of coronavirus in Europe. That's where I did my training, and seen a lot of feline infectious peritonitis cases, TZE, and other infectious bronchitis. One thing which is very distinct with the ADE is the ADEs primarily affect where the virus affect the dendritic cells, the macrophages, or myeloid cells. And as far as our understanding now, with a lot of cases we know from coronavirus, is it does not affect the point of contact in humans is respiratory epithelium cells, whether it's the ciliated epithelial cells or down in the pulmonary epithelial cells. So I think we can pretty much debate whether there is a risk for ADE or not, but I don't agree based on the data currently that there is any evidence of ADE, although dengue virus, where in humans there was ADE, but the dengue affect macrophages, correct? The feline infectious peritonitis virus, the highly virulent, that affects the peritoneal macrophages. So wherever we see ADE, normally it's the immune cells which get the first point of contact. The immune cells or inflammation in the lung due to coronavirus is the secondary to the lung damage or due to the cytokine release syndrome at the very terminal stage. So I don't necessarily agree that there is a huge risk of ADE with coronavirus, and you can disagree with me on that.
No, I don't disagree with you, and the points that you have raised are totally valid, and this is what you have discussed also in the review article that we have published, that there is no conclusive evidence so far, and as you correctly pointed out, that the virus so far has not shown that tropism for monocyte or macrophages, unlike FIPV. But preliminary data, very preliminary data, I should say, to be on the safe side, is that they did see some evidence in non-human primates with SARS, but not SARS-CoV-2, and they found some cytokine profiles in SARS-CoV-2 patient with poor prognosis that mirrored with what you typically see with monocyte macrophage activation syndrome. But this is all the information that is available. So which I totally agree with you is not sufficient enough to be concerned. But this is something that should be in the radar because the pathogenesis of the virus still remains to be conclusively elucidated or completely elucidated.
Absolutely.
Yeah, thank you, sir. That was interesting
So
point.
Dr.
Thank you,
Sethi,
sir.
just check with Dr.
Sir.
Scott whether he is available now. Scott?
I don't think he's there. We will move with the
Message from
next
Dr.
speaker.
Baljeet.
So, Dr. Sethi, can we have a one or two questions from students that I'm happy that students, they have some queries.
If you
Okay,
have
at
Can
least
we take?
have quickly one question.
Yeah. This is from Sir Asim Sahu from ICMR, NIV. And the student is asking whether there is any research conducted that conclude how fast mutation rate is going on in SARS COVID-19.
Yeah, that's a very important aspect, and there has been several publications on the accumulation of mutations based on the information from published data, and that also I'm speaking about when you communicated the review article, and that is almost more than 2 months. So a lot has happened after that, but there has been mutations, but until recently, those mutations based on the published data doesn't speak of antigenic drift, but recently variants have been reported which are a cause of concern.
Thank you, sir.
And one of the things is the virus has a large genome. So obviously, if the genome is large, it is more plastic and is vulnerable. But one thing is that the coronavirus is a proofreading mechanism, but on the other hand, it replicates very fast. So which may compensate the advantage against mutation that you get with the proofreading mechanism.
Thanks, sir. One last, sir. What could be the possible reason for the short-lived immunity elicited by the animal coronavirus vaccine? Why the immunity is so short?
I am not aware of any conclusive information available on that. If one of my colleagues may want to say something. But similar observations have been seen in animals.
Right. Thank you, sir. Dr.
Thank you so
Sethi.
much.
Thank you, Dr. Shovik Ghosh. Thank you very much. And you will be surprised these days, every day, online webinars are happening. Attendance is very thin, but today you can see more than 200 participants.
Yeah, I'm very impressed.
And this is encouraging. This is not encouraging for you as well as for us also. And simultaneously, this is being live on YouTube as well as the society's webpage, Facebook. So we can expect more people hitting this and watching all these proceedings. Thank you very much, sir, for an elaborative talk. Thank you very much, the organizer, the society is extending their thanks for such a nice talk. Thank you very much, sir.
Thank you so much. And I would also like to extend my heartfelt thanks to the organizing committee and the audience. Thank you so much for this opportunity. It was
Very
indeed
much.
a pleasure sharing this information.
Okay, let us move with our next speaker. The next speaker is, you are already familiar with him, Dr. Banu Pratap Singh. Dr. Banu is currently working as Senior Director Pathology at Gilead Sciences, US. He has graduated from GB Pant University, we know Pantnagar, India, and did his master's from Netherlands. Then he completed his pathology resident training at Royal Veterinary College, London, UK. During his residency training, he did internships and externships. You will be surprised that he had been to Bern University, Switzerland, University of California, Davis, Armed Forces Institute of Pathology, Washington, and National Institute of Environment Health Sciences. He had been a visiting fellow with Dr. Dave Malarkey's laboratory. Uh, Above that, he is a board certified veterinary anatomic pathologist, as well as board certified toxicologist and a fellow of International Academy of Toxicologic Pathology. Sir, I could not believe such a glamorous CV. He has over 10 years of industry experience in non-clinical safety of oncology and cardiovascular metabolic diseases program. He has a strong scientific interest in drug discovery, especially oncology and cardiovascular metabolism, molecular pathology, liver toxicity, development of animal models of human diseases, and chemical carcinogenesis. He has authored over 5 book chapters, 80 plus regulatory and research pathological reports, number of peer-reviewed publications, and delivered more than 70 presentations in various scientific meetings. Dr. Singh also served as a chair for various societies on education course committees, associated editor for drug and chemical toxicology. We are lucky to have today with us, and he will be speaking on COVID-19 vaccine, current global landscape and future perspectives. Dr. Banu, we welcome you, sir.
Thank you, sir.
Please go ahead, sir. So before Dr. Banu starts, for the participants, please keep on typing your questions in the chat box. We will take on your behalf. Thank you very much.
So just like Dr. Souvik, I would like to check if you can hear me and you can see fully screen mode my slides.
Yes, sir. Your screen is visible and you are audible.
Perfect. Well, thank you for the very kind introduction, Dr. Seri. I have to say, you know, I'm just I feel very honored to be on this platform with such a senior virologist and other experts speaking along with that. So it's actually I feel privileged and honored to be here. I also wanted to congratulate the Indian Virology Society for putting such an amazing program. This is my first ever interaction with Indian Virology Society. Hopefully not the last one, but I'm very impressed. And thanks Dr. Malik to connect me with this platform. I'm also try to stay as long as I can stay because this is a very interesting program, and I would like to learn from other experts here as well. You already heard Dr. Ghosh gave an excellent talk on the veterinary perspective on the COVID vaccine development. What I'm coming from I'm looking the COVID vaccine development from a different point of view, and I will focus. I stay very high level and you know, more from a commercial pharmaceutical side, industry and public health policy perspective. And what I will try to explain is where we are currently and what are the future challenges, because this is where, you know, I think we want to learn where currently we stand. I have to warn you, what I may say today may not be true in 2 weeks, because in this world, especially COVID-19, the information is changing so fast, and I think we are learning so much that the topic in the science is evolving day by day. So what I try to give you the most updated information available as of last week when I made these slides. So that's how much I can claim. But after that, the things are changing day by day. This is a very evolving field. This is just a disclaimer I have to give that whatever I say, I'm not releasing any confidential information from my organization, as well as most of the information is available publicly, and it's my view. It doesn't represent my organization's view in any way or any shape. So as I said, I will explain where we are, and then we'll talk about the vaccine landscape, current landscape. Then we'll talk about some of the challenges which I think all are facing. So this is all where it started. I think it's a little over a year now in Wuhan, which is a major cultural and education center in China, and is a major transportation hub. It's about 11 million people live in the city, and that's where the first case was reported. And by the time I think it was reported somewhere in the first week of December. And when they start isolating, by the time it was the end of December 30, that's where they isolated the viral sample from bronchial alveolar lavage from this patient. There is, based on the preliminary epidemiology data, the case was connected to this wholesale seafood market in Wuhan, in Henan province there. And since then, it's 1 year, 1 case, 0 case. And if you look where we are, this is the data as 1 week before. It changes every hour. This data here from Johns Hopkins University. As you can see, as of now, the virus is impacting 150 countries in Asia, Europe, North America, South America, Africa, and Oceania. So almost all the continent, you can imagine. The virus is, in 1 year, it has gone so far. We know very well this translates human to human, and that has been confirmed. In the beginning, there was some speculation on that. And there are currently, as we talk, over 95 million cases worldwide. And unfortunately, we have lost 2 million people worldwide related to COVID-19. That I think, if we look back 1 year back, when we were just hearing in the news, but as of now today, I can speak. I know some people who are infected, or some families who have lost their loved one because of COVID-19. So this has impacted at a global level to us. In the US alone, more than 400,000 people have died so far. So this is unprecedented. Within a year, we go from 1 to 25 million. This is a fast spreading virus. Then, as you recall, when it all started, we start trying to manage government across the globe. They try to come up with different ideas. I mean, the first thing was lockdowns, mask, social distancing, government mandated. It's very strict procedures were followed. Then, personally, we tried to learn to live. We start working remotely. We got now expert in Zoom meetings and Zoom calls. I have to certainly learn, and so do other people. So it affected a lot. Meanwhile, as soon as the cases were raising up, the pharmaceutical companies and academics and scientists all around the world, they were trying to come up with. We knew that the vaccine is going to take time, so there was a lot of focus on developing therapeutics or medicines to treat coronavirus. The first drug which was approved was from one of my organizations, Gilead Sciences, that were remdesivir. The trade name is Bacthlori, and this was approved in October. And the reason why it got so fast compared to other drugs, because this drug was already tested in animal models as well as in some humans. Originally, this drug was aimed for Ebola virus. So when based on the clinical trial, this drug was the data was submitted, and FDA gave first conditional approval, and then later it got a full approval, not only in US but in other countries as well. The drug is also available in India with different name there. That was the first drug which came, and then I think right around that time, we also learned the serum from the people who have been infected, the convalescent serum, that can be useful for treating the patient as well, and that was approved as well. Around June last year in UK, dexamethasone, which is well-known drug, it was approved to treat the severely ill patient. And the way it was working, it was trying to inhibit or stop the inflammation in the lung, and thus trying to reduce the mortality in this terminally ill patient. Then after that, a lot of attention got last year, I think in November, on monoclonal antibodies. This is the same concept as designing the protein, attacking the spike protein in the virus. And as of now, Eli Lilly and Regeneron, they have conditional approval for monoclonal antibody for COVID-19. And this has got a lot of attention because if you remember, our beloved President Trump, when he got sick, he got in the news, and as you recall, he was treated with Regeneron antibodies. So as you can see, it got a lot of attention in the media as well at the time. Nevertheless, I think these are the tools we have currently available, and there are some other drugs as well all around the world which are either in trial or getting conditional approval. But these are the drugs for the patients. These are not to stop the infection, to treat the sick patients. Meanwhile, if you look what's happening around the world, we almost halted. I can hardly see anybody traveling by plane these days, only in case of emergency. No meetings, and our life schools are locked down. My kids have not gone to school for almost a year now. There is new concern with the new variants. UK had to put some new measure recently in November to have another phase of lockdown. So this is an ongoing problem. The economy has, impacted all across the globe, and some of us are lucky who had job and who are able to manage work remotely. But there's a lot of sectors, the apparel, the demand for clothes have gone down. So some of the companies have filed bankruptcy. If you look on the right side, we are not driving that much. So the oil and gas industry has suffered. So that and some of the stores have closed for good. There's no business. So this is also unprecedented in 2020. A lot of these sectors, business sector, has got a huge impact. Some of them are trying to recover with the help of the government, financial help, but still restaurant business and a lot of businesses, they have not come back yet. And there are still lockdowns in place or restrictions in place. So as you can see, without vaccine, I don't think we are coming back to normal. That's very clear. And then you ask the question, why does it take time to make vaccine? Why can't develop fast? Well, there are several roadblocks, as we know from manufacturer or the pharmaceutical side. You have to demonstrate both efficacy, means the vaccine is efficacious, as well as it's safe before you can put you can go commercial and put in a large number of people. Is there a process? And even if you discover a vaccine today in the lab, we're talking to millions of doses. So we need to, whatever process or whatever vaccine you have, it has to be in a way that it can be scaled up that it can meet the demand. Otherwise, it's not going to be any good. We have to maintain the quality. We are injecting something in our body, so the quality has to be, there should not be any compromise to the quality of that vaccine. As you know, the vaccines, they often use the raw material, intermediate, and excipients. Those cannot be compromised as well, because they can cause some serious reaction to and cause issue with the safety. Most importantly, who is going to invest the money? I mean, research requires a lot of money, whether it's in academic or in pharmaceutical. So obviously the government has stepped up across the globe to help the pharmaceutical and the labs to discover or develop these. But these are the kind of challenges, to make a vaccine which can be available all across the globe. The next thing is the timing. We don't have time. 1 year, we already has seen so much of impact all across the world. Typical vaccine, as you know, it goes through sequential phases. We have discovery phase, then it goes to the animal model, then you submit IND, the investigational new drug, to the regulatory agencies. They will allow the human trials. And then it goes into phase 1, phase 2, and phase 3. So as we move forward to various increasing number of people get tested. If you look how we have worked around on those vaccine development for SARS-CoV is by designing, either skipping some of these steps because we already know based on the other coronavirus or previous experience. And then if you see here, it's overlapping. So as the phase 1 is not completed, based on the interim result from phase 1, the phase 2 has been started. And phase 3 is also starting. So there's a lot of overlap to try to compress the result. At the same time, you cannot compromise the scientific data or the scientific integrity. The government agencies, typically they take about 1 to 2 years to review the whole package and the biological licensing application. They're trying to review it very fast in 1 to 2 months. So everybody is trying to help, and we have made significant progress in bringing these medicine. So now we are about 10 months to 1.5 years. You can start hearing there are different vaccines available. And this is where we stand as of now. We have 41 candidates in phase 1, 22 in phase 2, 20 in phase 3, 8 which have been conditional approval, and 2 which have been given full approval to be used for general population. So I think this is an impressive progress given the complexity it takes to design a vaccine. And I apologize for the very busy table here, but this is where we stand right now in terms of the approved vaccine, or the one which you hear in the news quite a lot these days. So there are mRNA-based vaccine. The one is from Pfizer, Comirnaty, and this has been approved in a number of countries, including US. And as of now, in US, you can get these, although in some only restricted people for restricted use right now. Same thing with Moderna, which is a company, is also come up with mRNA-based vaccine. It has been approved in several countries. The other vaccine platform is adenovirus vaccine, which is AstraZeneca and Oxford. This is the one which has been approved, has given approval in UK and India. This is with the name of Covishield. So it's the Serum Institute of India, which is manufacturing this based on the technology developed from AstraZeneca. There are other countries which have come up with vaccine, whether they are inactivated vaccine or viral vector vaccine. There is one which has got a lot of attention. It's coming from Bharat Biotech, and this is an inactivated vaccine. So this is not an mRNA vaccine. Inactivated vaccines are known to us, while the mRNA vaccines are fairly, new technology in terms of vaccine development. So this is where we stand. There is obviously a lot of efforts to scale up and produce millions of doses for these vaccines. A little bit about the mRNA vaccine. But before we learn about what the mRNA vaccine is, we need to understand what the virus is. And Dr. Ghosh already explained about the virus, and I will go briefly. This is the major protein, the spike protein on this virus, which attached to the receptor on this epithelial cells. And this has been a target of vaccine, whether we are trying to do mRNA vaccine or peptide-based. So this S protein is what we try to synthesize when you give somebody the mRNA vaccine, which is lipid coated in the liposomes. It gets into the cell, and this mRNA will translate and make the spike protein, which will be expressed on the cell surface here. From there, it's been recognized by your immune system, and it can have either the cell-mediated immunity, which is through the cytotoxic T cells, or the antibody-mediated immunity, which is through the CD4 positive T cells. Both of these cases will lead to the immunity for these viruses. One thing, the advantage is for mRNA vaccine is they will stimulate your immunity by both cell-mediated as well as the antibody-mediated immunity. And they are considered safer, and they will provoke the strong immune response in the body. Another platform, which is an AstraZeneca and Oxford vaccine, is the adenovirus vaccine. Just like the mRNA vaccine, the main idea here is to insert the gene for spike protein in adenovirus, and that's being inserted in the cells, recognized by the body, and that will provoke the immune response. And there are two ways you can have these adenovirus viral platform. The one is the replicating viral vector or the non-replicating viral vector. The vaccine, which is from Oxford and AstraZeneca, is a non-replicating viral vector. That's why I think you have to, the replicating virus, viral-based vaccine, they don't require booster, but the non-replicating generally require the booster injections. That's why we have to have 2 injections for this vaccine. So, you will hear a lot in the news these days. Is it we are going very fast? Some people are saying we are not going very fast. Some people are going, we are going very fast, and we are not doing a good job in following the traditional safety model, safety efficacy model. And I think there is a both side of the story. If we want to accelerate or go fast, there is a scientific and social value. There is a need for this vaccine. Everybody knows that. And the sooner we get it, sooner we can get back to the normal life. We can save many lives. We can improve many lives. It will raise our confidence, and we can start, going back to the normal. But there's also risk. What about if these are not have been vetted properly? There's a uncertainty about efficacy data, because a lot of time these being approved on limited phase 1, phase 2, and phase 3 data. But is it fair to have those shortcuts? And the question is, should we wait for 2 years to get the perfect data, or we should go ahead with whatever we have? And this is a not only just a scientific question, it is a public health policy question. It becomes a political question as well. It is, it's a very debatable topic, but I think in the interest of the public health, somebody has to make that decision and move on. But the important thing is we need to be transparent and tell people what it is, what they're getting. We should not overpromise anybody. People should be aware what the risks are. People should be aware how much efficacious those are, because unlike traditional vaccines, these are going fast. These are being developed in a very fast way. So you will hear about different vaccines. There's the mRNA vaccine, there's an adenoviral vaccine, there's a Moderna vaccine, AstraZeneca vaccine. Are they different? Which one should you get? Well, that depends what you can get. Right now, we have such a shortage that you have to get into the line or wait to get the vaccine. So I would get any vaccine which I can get first, allowed to choose. But right now, it's just whatever we can get to back to the normal. But there is not much difference in terms of vaccine, whether it's in terms of efficacy, they're pretty much efficacious. 95, 94, even 70% is a very considered good efficacy for a vaccine. They all of them require 2 shots. Some of them require at 3 weeks. So you have to go back to your doctor to get another shot to get the full immunity for these. The important difference is how they are stored, and that makes that's a very important distinction, because if you I grew up in India, so I know it's very hard to maintain minus 70 degrees Celsius transport from Delhi to the town where I live. It's how do we supply millions of doses at minus 70 degrees from in the hot summer from one city to another city or one place to another city? Not only India, there's even in US in areas, it's a nightmare, logistical nightmare to maintain that temperature. So obviously the 2 to 8 degrees Celsius, which is much more easily manageable vaccine to carry around and store, will be more suitable for certain countries than other countries. The vaccine is not really, these mRNAs are very fragile, as we know. If they get exposed to the heat, these vaccines are not good anymore. So that's, I think, is a major difference if you look at different vaccine. As far as the safety profile is concerned, they're all pretty much fall in the same spectrum. Most of these, you will have injection site pain and fever, muscle pain transiently. Not everyone gets it. Some people get it. And these are more sign that your immune system has been turned on. It's nothing wrong with the vaccine itself, because most of the vaccine, they do have similar kind of reaction. As far as giving those vaccine to pregnant or the mothers who are feeding their babies, we don't have sufficient data. These to see these kind of effect, the studies need to they take a lot of time. Those are underway, but we don't have sufficient data to say whether this will be safe or not. This is a snapshot. So this is as good as of today. This is how many people have been vaccinated around the world. 60 million people, 60 million shots have been given all across the globe. There's about, out of these, I would say about 2 million. So this becomes about 3 doses a day. That's a lot of vaccine to consider. But a lot of people in United States has been vaccinated. Then in China, Russia, Western Europe, India has started vaccination, as we know. Some part of South America, in Brazil and other countries, Canada. So there's a lot of countries which we have started. All of these, the vaccine which have been approved, they have been ongoing vaccination there. So this part of the talk, I will try to address the challenges. And these challenges are real. They may sound a little bit hilarious for some people, but I would say we are also dealing not only with pandemic, but there is a pandemic of disinformation. And this is very, this is not productive. in the time of pandemic. We need real information, not made up information. And I think you probably have seen one of these in your WhatsApp or Facebook. I can almost guarantee you, I got all three of them. And it's for a scientist like many of us, we might ignore it, but we have to understand this information is circulating all around the globe. Not everybody, you know, is thinking that it's a real or not, and it can have a huge impact on people. You know, whether they believe this infection or not, whether they want to take vaccine or not. So this disinformation has a real impact. What we have to also understand, when most of the people get these as a social media message, whether the Facebook or WhatsApp, they forward it to 5 other people. And I myself have been victim of so many forwarded messages on coronavirus, vaccine, medicine. But you have to understand, the person who is forwarding it, he's not really trying to give you the real information. His interest is how many likes I will get on my Facebook post if I put this information. That is the person putting there. I think he's thinking very less about whether this is an accurate information or fake information, but it's just most like how many likes or how much popular this post is going to be. That is, and that is a dangerous situation in the middle of the pandemic. We don't need misinformation. This will misguide people, and this will have the impact. So what we need is reliable information and science-based decision making. That's where the scientists and the public health and all of us come and help the society in general to guide them and to provide them, to filter the information and to give them right information based on the science, not based on publicity. And this is social media, but our politicians, it doesn't matter which part of the world you are, they are also part of this misinformation campaign. I don't mean to point one person or one political affiliation, but in general, these kind of statements, whether you heard about hydroxychloroquine, which got a lot of attention in the beginning, and later FDA found out it was causing heart attack in a lot of people, and that was dangerous because people did not realize how unsafe that drug was, and it got stockpiled and whatnot. Then there was some promises made with another name, and then even the vaccine is getting politicized. Whether which I want this or not. Everybody knows about this kind of statement. But I heard somebody say something on the TV one day. This is the fact. If we forget everything, the virus doesn't care about our political preference. The virus doesn't know which party or which country, which race or which religion you belong to. It's going to affect everyone. So we should remember that. So these political statement may be based on somebody's opinion, but the virus doesn't care what your political preference are. So I believe it should be doctors, scientists, the people with the first-hand information. They should lead the public communication in a transparent and evidence-based way, not politicians. That's my personal opinion. Science should drive the health care policies, not the politics. And there has to be a better coordination between the news media, the scientists, and the government. And again, keep the science in the focus, not your political preferences. And that's going to help public health tremendously if we focus on that. Second, so we have first challenge is the information we are dealing with. The second challenge is also real. Now that we have vaccine coming out, is how you will distribute it to everyone, not only in one country, across the globe. And there is funding problem. How are we going to fund it? And a lot of governments are being very generous in giving money to the manufacturing and trying to scale up and giving the access to the people to the vaccine. There is a supply and logistic. we just talk about how you're going to transport something at minus 70 degree, 2,000 kilometer or 3,000 kilometer. That problem is a real, how we'll make accessible to everybody. There's a communication and trust. I think I talked enough about it. How do we make people believe that this is a real problem and we need a real solution for it? A lot of people still don't, they have their own theories about this virus and the infection, and we can ignore them. But if those people don't get vaccinated, or they don't, then the virus will be still here forever. I think we need to have a large population believe, this is a real virus. WHO has stepped up and they have put a framework, because WHO recognized that we need to have the vaccine globally available, and they have a program which is an ACT accelerator. This is a global framework to make vaccine available for majority of the countries across the globe. In phase 1, the countries are going to receive the vaccine based on their population, and that AstraZeneca Oxford vaccine, that is the one which is developed by Serum Institute, it's an effort of ACT accelerator. First phase, there will be frontline worker in health and social care setting. They will be given vaccine. And I think as of now, those are getting vaccinated. Then there is a high risk adult, the people who are 65 plus, underlying heart condition, blood pressure, they will be get vaccinated. And then there will be phase 2 where more or less healthy adults, they will be given the vaccine. So most of the countries are following the phased approach to start vaccinating the people, and there will be always a buffer, just for a humanitarian deployment and in case of an emergency. Lastly, I think this is, again, Dr. Ghosh mentioned this. This is another challenge which we are dealing. We haven't controlled the first virus. Now we have, I just put it 3 variants, but there are more variants coming up. And I would say, there's been variant which has been recognized in the UK. That was the first one in September. And now even this variant has been reported in the US. And I'm sure this is probably in other parts of the world as well. Another variant came from South Africa, and the one from Brazil that was reported at the airport in Japan from people traveling from Brazil. The reality is, I think with the data and the science is going on the research which is going to understand what it does. We know the virus from the UK that has a high transmissibility, and it's affecting, but it is responding to the vaccine. So as we speak, there's a lot of work and research work going on these variants to understand how will this impact. And the consequences of these emerging variant, it could be manifold. They might spread fast, so then we have another problem. They might not respond to the available therapy or available vaccine. They might not respond to the diagnostic test, and that's even more challenging. If our test, the diagnostic test we have, they fail to detect these variants, they can evade the vaccine, and that can be a problem as well. So hopefully, I think I try to convey you that this is a global problem. It's not a national problem, and the global problem will need a global solution. We cannot be, focused on one town or one city or one country and hope that this virus goes away. It's not going to go away. It's a global problem, and we need to have a global solution for it. There's an optimism, there's a hope, and there is a good news. The good news is the vaccines are being developed, and the getting scaled up at a high capacity and getting distributed across the world. It's known that the vaccine is going to be in short supply. We cannot vaccinate everybody in the beginning, and governments are taking phased approach so that the people who are the most vulnerable, they get vaccinated first. And what is still, there's a fear of these new variants and hopefully the science will drive our decision on, whether these variants can be managed with the available science. So with that, I really want to thank my wife and kids to give me some time so I could have work and focus on my slide, and Kamna also she also works in pharma and helped me with some of these slides as well. So thank you for your attention, and I would be happy to take the questions now.
Thank you very much, sir. Dr. Ratan, do we have any questions?
Yeah, thank you, sir. We have one question from Kishore asking how vaccine overcomes the mutations of COVID. Dr. Bhanu.
Can you repeat that question? I didn't.
Question is how vaccine overcomes the COVID mutations.
We don't know about it. I think the vaccines have been designed on the spike protein. So if the mutations are in the spike protein, theoretically, we can say the vaccines may not be effective, but I would not comment. I think this is something which require research work and focused research work to address that question. It may or may not be.
If I may ask a question with your permission.
Sure.
I have one question for you.
Yes, please.
Yes, please.
Dr. Souvik? Dr.
Or
Souvik?
should I?
Yes, if you want to ask, yes.
Yeah, okay. Dr. Singh, that was an excellent presentation. Thank you for the informative presentation. I do have a question with regards to the
Please go ahead, sir.
mRNA. I do have a question with regards to the mRNA vaccine. One of the concerns of the mRNA vaccine is the thermostability. So we do work on RNA viruses, and previously I do remember that when I have to send RNA samples to some place, we need dry ice, and it was a nightmare. But now we have certain technology, like we have some tubes available called Gentegra tubes, where you can add the RNA, you can desiccate it, and you can transfer the RNA at room temperature, and the RNA remains stable for long duration. So are you aware of any research that has been directed to increase the thermostability of these mRNA vaccines?
So that's a good point. I am aware of that. One of the major concerns, Dr. Bose, is any of these manipulation, let me call these manipulation to stabilize these mRNA. You're adding chemicals, and then we don't know the consequences of giving those chemicals in, we don't know the safety, how it interferes with the efficacy of the vaccine, how will it cause any severe reaction in the patients. So I think there is a, I don't know, because a lot of these are proprietary information, but I'm aware companies like Moderna and other companies, they are trying to work on it. But the agencies, the government agencies, when they approve the vaccine, they look not only the vaccine, but whatever the component is in that vaccine. And they look at the safety profile of those as well. So I'm pretty sure that there are other companies which are coming along the way, and we will hear more progress. But the more data is needed to characterize the safety of these adjuvant or any of these chemicals which you will be adding in the vaccine.
Thank you, sir. And I have just a comment with your permission about the variants. Yeah, the mutations are indeed a concern. And as you mentioned in your slide, that there is no conclusive evidence in humans that show that certain mutants may have increased transmissibility, although there is evidence, preliminary evidence on that. But the variant that has circulated and is predominant in the world, that is the D614G mutant, studies have been conducted in hamsters, and it has shown that the D614G mutant is more infectious in experimentally infected hamsters compared to the original strain. So I think we need more studies in humans with regards to the impact of the mutation on transmissibility and vaccine efficacy. Thank you, sir, for the excellent presentation.
Okay, Dr. Bhanu,
absolutely, and I would say I'm not a biologist or any expert claiming to be expert in the biology. There are much more expert people here on that topic. But I do agree with your comment.
I have one more question from participant, Pankaj Kumar Yadav. He's asking you how to combat with wrong information spreading around regarding the vaccination and its adverse effect.
Oh gosh, you're asking the political question to me. That answer goes off the record. So I would say I'm not answering this question as a scientist, but as a general thumb of rule is any WhatsApp message. Really, sometimes you feel like you have to send it to other people. Go on Google and put that message and put a word fraud check. And I can almost guarantee you will find somewhere this information has been debunked. And educate yourself first before trying to spread that information. Otherwise, you will become part of that misinformation campaign. And that's all I can say. I don't really have any other influence on that.
I think also,
I can control myself only. Yeah.
Go for the scientific information rather than, popular thing is spreading.
Absolutely.
That's
Absolutely. If we look around, I'm not saying that every family has is an expert on COVID, but we have friends, we have family, we have relatives who are doctors. And I think those should be our point of contact to seek the information rather than WhatsApp or Facebook. Those are the last place I want to go for my medical information.
Thank you.
Thank you, Dr. Ratan, and thank you very much, sir. The organizing committee is thankful for sharing your thoughts and elaborating journey of initial efforts for the development of vaccine to the present data we have, as well as posing future challenges associated with safety, transport, and delivery of COVID-19 vaccine. Sir, I personally feel the development of vaccine in such a quick fashion shows us the power of collaborative science, which we call teamwork, for the betterment of society. However, these development at the same time raises certain questions about the safety and efficacy of vaccine. Thanks for clearing these basic questions, which are presently in the minds of millions of people across the globe. Once again, we are thankful to you, sir. And as you earlier said, the Indian Virological Society as well as College of Animal Biotechnology will be looking forward for more interaction with you, sir, in future. Thank you very much.
Thank you.
Thank you. And I think you are doing an excellent job. And this webinar in itself is a good example. I think the more we talk to people, the words get out. And I think the more people will believe in science rather than misinformation. So thank you.
Thank you very much. So our next speaker is Muhammad Izzat Jovalti.
Yes, good morning, everybody.
Good morning.
Dr. Jovalti, thank you very much. And Dr. Jovalti is presently Associate Professor, Genesis Science Center, Department of Medical Biochemistry and Microbiology, Uppsala University, Sweden, and International Research Collaborator, Duke One Health, Duke University, US. He has been awarded a scholarship as a visiting scholar in the Department of Cancer Biology, Duke University Medical Center, US in 2006, where he studied effect of sweeteners on gut microbiota in mice. He is passionate about virology, virus pathogen discovery, analysis of virome and microbe, zoonotic pathogens, including but not limited to influenza viruses, coronaviruses, and Newcastle disease virus. Today, he will be speaking on SARS-CoV-2 and emerging pandemic threats and One Health issue. Dr. Jovalti, please.
Just to add, Dr. Sethi, Professor Mays and me both worked with Professor Goel, Sagar Goel. So
Okay.
we got connected few years back from Dr. Sagar Goel, and I found Muhammad Professor Mays, what I call him, because his name is little long for me. Muhammad Izzat Al Jalte. I call him Professor Mays. And we both are working since long now with the connection of Professor Sagar Goel at Minnesota University.
Sir,
Yes.
the moment we discussed about him, you always said Dr. Mays, and I was bit reluctant to say him Dr. Mays. Now I understand why you are referring him. Thank you very much.
Please.
So, Dr. Joelty, please.
Okay, good morning, everybody. First of all, please allow me to thank Professor Yash Singh and all the group. That's a fantabulous researchers. And this is very timely webinar on COVID and emerging threats. So again, I would like to express my thanks to Dr. Singh for his honorable invitation. And as Dr. Singh said, Dr. Sagar Goyal, my mentor previously in University of Minnesota, he is very nice and helpful, and I'm sure most of you know him. So we would like to also thank the opportunities that connect scientists together. Without connection and collaboration, this wouldn't have been achieved. So today I would like to discuss and emphasize about the emerging pandemic threats and One Health issue and its connection to the emergence of SARS virus. As of you know, emerging viruses are a continuous threat to human health. And the objective from my talk will be focusing on One Health and emerging viruses and the viral zoonosis, as well as the emergence of zoonotic SARS-CoV-2. And my research on pandemic threats using One Health, I will provide some information for you. So now this is the world we are living in. As of yesterday, 97 million plus cases of COVID and 2 million plus total deaths. These numbers are just yesterday. So the numbers are continuously increasing. And this is the initial images, snapshots from Johns Hopkins Center. You can see that the red dot was somewhere in Wuhan, China. And after a few months, and this is on February 2020, and then after, then in April, the whole world get the virus. And now this is the situation we are living in. Every single corner on this planet is reporting or had reported a case except Antarctica. So what's going on with viruses and virus infections? Then let's have a global view. This is our planet where we live. There is no planet B. Now it's only the Earth which we can live on, every single creature. So taking the timeline of newly emerging viruses are there long time ago. The evolution of such viruses is a complex story, and there has been a lot of viruses that has been reported to infect humans, including West Nile Ebola, HIV, and Nipah. And somehow recently in the 19th and 20th century and 21 now, SARS-CoV-2 from pets, and then MERS coronavirus in 2012. And then more recently, as reported last year from Wuhan, China, SARS-CoV-2 has emerged in 2019. However, still research, probably the virus may have been there before that date. We don't know yet until solid evidence of science is coming up. So these are examples of wicked problems. And I'm sure most of you know what wicked problems are. Wicked problems are social problems that affect humans, and they are characterized by being difficult to clearly identify, and a lot of interconnections, and very complex and socially complex, and affect the humans, and they have unforeseen consequences. So examples of such wicked problems are infectious diseases, and also food security, and antimicrobial resistance. So I will highlight from this presentation about emerging infectious diseases and the relationship with SARS-CoV-2. And while these problems are unique, they may be interrelated or interconnected. In the G20, all such problems has been discussed over the past years, and to combat antimicrobial resistance. And the word One Health has been reported previously in a G20 summit, and also the importance of health and health issues, and the fighting against antimicrobial resistance and against pandemics has also been discussed in another more recent G20 G20 summit. And also the importance of the health risk and health security, including AMR, it has been the question in a more recent health summit. So health is a prerequisite for sustainable and inclusive growth. And last year in the G20 that was recently led, it was the first G20 summit to be virtual summit due to this coronavirus, and it was an extraordinary summit due to the coronavirus pandemic and the social and the physical distancing, and it has been held virtually, where the leaders has discussed the coronavirus pandemic. So we all living now in this hard times and situation. We have to tackle this problem in a more efficient way. So the management of such health risks and health security, outbreaks and other health emergencies, it's a very complex and a very it's a problem that has a lot of angles we have to look. It's not only we look to the human health, we have to look into animal health and environmental health, and that's all as will come later under the One Health issue. And the global risks that target or challenge humans include also in the spread of infections. And this is a very nice report from the World Economic Forum on the global risk, and infectious diseases are there as a risk that affect humans. And this is show to you how these issues are very complex and are interconnected to changing climate, to poverty, to other issues, aging and the populations. So these are examples of those wicked problems. So One Health is an approach or a science policy that tackle these complex problems and try to solve it, and to reduce and mitigate the future virus pandemic threats. And using the One Health approach, science now has proved that this partnership is very important. So what is One Health? So the more people we have, the more animals we have and carrying more diseases, this is a very perfect viral melting pot or viral production or production plant. So One Health, we have to consider an outbreak somewhere is an outbreak everywhere. So humans are living on this planet in an environment with livestock, with wildlife, and here we are. We are in a One Health issue, and One Health will also tackle these kind of issues. So the approaches to these wicked problems is the One Health and One Health triad, meaning healthy people, we will living in a healthy environment with healthy animals, then we will have healthy people. And most of you know One Health is an umbrella that combine all the disciplines under this One Health to tackle the issue, and it also study the infectious diseases and how they emerge. So as you know, emerging in zoonotic diseases which transfer from animals to humans, and it's estimated that 2/3 of those human pathogens are of animal origin, and most of them have intermediate host also from wildlife, and the number of emerging infectious diseases has been increased due to the close proximity and the more animal production and the more contact with animals and more population. So zoonotic diseases, again, it's estimated that 75% of from 61 to 75, all of these are zoonotic and causing a threat, a continuous threat to human health. So this current novel, now it's not a novel, and this current coronavirus pandemic has proved that animal health and human health, and as well as environmental health, when they are interconnected, this is require a One Health issue to tackle this challenge. And most of us remember the avian flu, the MERS coronavirus, and the swine origin pandemic H1N1 and other variants, H3N2, and the Ebola virus. And here we are in the SARS-CoV-2. This is a very nice model showing you how these pathogens are, the genesis of these pathogens in the wild or in the animal population until they're transferred to humans and make an efficient establishment of a human-to-human transmission. So we have to study zoonotic pathogens, either bacterial or viral, and their respiratory or enteric, vector-borne, and in any cohort of animals, such as wildlife or domesticated, and in urban or rural. So it takes a few years until these pathogens jump and spill over from those populations, wildlife, pets, into humans. Where are and how we can conduct the surveillance for future pandemics? This is also a nice model. We can look into wildlife virus focus. For example, we look into the ecology and intensive virology, and we do genetic studies. And then the study may have a human focus. We look into patients, the epidemics or the outbreak investigation. So what will combine these together? It's One Health. So One Health will look into animal and human at the joint or at the interface. For example, we can look into pneumonia etiology. So predicting zoonosis, we will predict zoonosis using this One Health approach. Most of zoonotic diseases are naturally, and here between brackets naturally, because as you may know, some of these zoonotic diseases may be deliberately they may have a deliberate origin. So these images showing you the interaction between humans in some parts of Africa and Asia with the wild animals, including bats and non-human primates. And this close proximity with those animals definitely will ignite some of these outbreaks. And there are established programs on the preventing of those emerging threats. Some of these programs, such as the PREEMPT program, which aim to the investigating of the virus mutations and cross-species transmission, and also the PREDICT program or the VIROM program. Most of these are well established, preparing for the next pandemic. And here, showing the timeline in brief of the One Health. One Health is a long time term since the 1800s, when the term was coined and the zoonosis was coined. And then now we are in 2020, and we need to establish the One Health on a global scale, not a regional or national scale only. And as we have mentioned, the healthy animals and healthy environment will definitely make humans more healthy and more healthier. And this all showing you that there is no one discipline can tackle such problems alone. We have to cooperate and collaborate, and there has to be interdisciplinary approaches. And no one single agency can tackle those infectious diseases. So that's why One Health, it's a holistic and interdisciplinary and multidisciplinary, multi-sectorial approach of challenging or tackling these health issues. And here showing you that when we look into an agent, where this agent is found in an environment, or what is the host of this agent, and then we establish the research programs using the One Health approach. So these all showing you the global change that favor the re-emergence or emergence of infectious diseases. And then we look into the disease origin at the boundaries or at the interface between animals and humans, animals and environment, and human and the environment. So we tackle all these issues. And here, these are recent examples of those emerging, focusing on respiratory viruses. We have the SARS-CoV-1 in 2003, 2004, and then we have the influenza 2009, the swine 1, and then we have in 2012 the MERS-CoV-2 affecting 27 countries with the numbers now. It's more regional in the Arabian Peninsula, specifically in Saudi Arabia, and then in 2019 we have this SARS-CoV-2, which is now the numbers are definitely now more than 90 million. So the One Health approach is a collaborative and multiple discipline approach. We work locally and working nationally or globally to attain and to achieve optimal health for every creature, every population on the planet. So we study human and animal diseases where the population mix, where there are interactions, such as swine populations, camels, most of you know, and live bird markets, as well as other domesticated animals and in far area, as we will see. So a lot of examples are there: influenza, arboviruses, and Rift Valley, Crimean Congo. A lot of diseases has been studied, and still we are looking for more studies on emerging infectious diseases at the boundaries of animal, human, and environment. So wild animals and epidemics, how are wild animals linked to an epidemic? Definitely wild animals, a lot of them hasn't been explored yet. And recently we have published this nice article. I would encourage most of you to read about from SARS until COVID, where we reach this current pandemic. And this recent article has been recently cited in the WHO interim guidelines on the SARS-CoV-2. And so from the Wuhan seafood market, where this COVID pandemic has been ignited, so there was a lot of animals there, and science hasn't yet determined the intermediate host, although some studies have showed up to that pangolins or some other animals may be the intermediate, but yet there is no definitive approach to the definitive intermediate host for the SARS-CoV-2. And this is a list of the animals that were sold in that market. So, and here we hypothesized that this virus is now well known that it's of bat origin due to the sequence similarity with bat viruses. And one fact that we know about is still much we don't know, and here it's this was last year in February, about the same time we start this idea about this article, and now after 1 year we're still looking for the intermediate host from this zoonotic perspective, and still research will determine and how our wild animals are linked, because of these wild animal populations contain a lot of viruses, and then some of the viruses start to jump and if it crosses species. a lot of wild animals has been reported to be present in the market at that time. And it's not only in China, it's everywhere. It's in India, it's in Egypt, it's in a lot of countries, Southeast Asia, even in the United States and in New York. The live bird markets are causing a lot of problems, and there has to be attention from the science point of view on the animals that are present in those markets. And the sequence similarity has showed the similarity of this virus to bad viruses. And here, the evolutioner of SARS-CoV-2, which is responsible for this pandemic, indicate that it has been circulating somewhere unnoticed in bats for decades, because the virus definitely is there, but we haven't got the attention to detect the virus. And this One Health projects will predict those pandemic threats. And this is another example of the animals that were present in the market. And this is summarizing the MERS story, and it's a similar story in bats, but we still don't know about the intermediate. And then it's come to humans causing this disease, COVID-19. And this is civet cats, which were responsible for the SARS-CoV-1. So also MERS from camels and the habits and the culture. So we have to protect humans and focus on zoonotic diseases, because healthy animals and healthy environment will definitely give us a healthier humans. Other similar examples are the influenza viruses, avian flu. This is a very nice photo from the 1919 palette on the North Carolina state of birth, showing one person has a cough or sneezing, and then it can infect millions of people surrounding. And this is what's going on now. We have these devastating effects and panic and fear among different population until today, until we find a concrete solution for the COVID pandemic. So viruses as a global infectious challenge, viruses will continue to be a threat to human health. And they are a major global health threats, and we have to tackle those. This is very nice photo showing you viruses go viral. An outbreak somewhere in any country is an outbreak everywhere. We have to work on a global scale, not only nationally. We are living in a very interconnected world, and that's showing you in Wuhan, the virus is spread to all corners, as you have seen. This summarizing the emerging viruses, as I told you, and now we will have to add COVID-19 and SARS-CoV-2 to this chart. So we are being challenged with a lot of emerging and re-emerging infectious diseases. So example of those are, some of them are very challenging and very devastating. And as we have mentioned, we are preparing using those pandemic predicting programs such as Predict, Virome, and the GLASS for the AMR. Also, due to this travel, for example, an outbreak in West Africa about monkeypox or Ebola, it can spread to the other parts of the world. Also, similarly, these are the not the flyways. Actually, these are the flyways, but this is when the emergence of antimicrobial resistance about carbapenemase producing and NMD, New Delhi metallo-beta-lactamase. Most of you remember this. By the interconnectivity of human activities, the AMR spread everywhere. And also the Zika virus pandemic, somewhere in South America, and then spread to other parts of the world. And also the Zika, and here the yellow fever and the monkeypox. And now this is caused of what? This is due to the spillover and the cross species of those viruses that are present in the animal population, for example, bats, and they cross to other animals. And here showing you the coronaviruses, as most of you are known, these are large family of animal viruses, and they mainly in bats and rodents, and most of them human coronaviruses which affect human are from bats. And also these such as SARS-1 and then the swine acute diarrhea syndrome, recent one, and the MERS, MERS, and now we are challenged with SARS-CoV-2. This is, most of us know this now about how the MERS is transmitted from pets to camels and then to humans. And this due to we have to look into from a veterinary domain and to look into zoonosis from a veterinary domain and from a One Health perspective, because as we said, One Health is a challenge, is an approach, is a measure that we look into the disease from different angles, from environmental, from human health, and from wildlife and domestic animals. Through this research, we will tackle these wicked problems, as we have mentioned previously. And the interest in One Health is widespread. There has been 65 institutions worldwide who has adopted the One Health program. And I hope taking this meeting and this webinar that you also have a One Health program in your colleges and nationally. And also there has been 68 almost or non-academic organizations who have a One Health program. And so what is the next wave of pandemics? We are now challenged recently emerging viruses, and now we will add SARS-CoV-2 to this list, and we don't know what's coming up next year or this year. A lot of viruses are coming up. So in brief, the following slides will focus on my research from the microbiology and virology research group, where we look into influenza ecology and surveillance. Giving you an example, we also tackle those diseases and emerging threats. We conduct research on viral zoonotic diseases. We focus mainly on influenza and rotavirus using a One Health approach. And also we focus on antimicrobial resistance using a One Health approach at the human-animal environmental interface. This is showing you we are in the field in South Africa. We collecting samples from wild birds and from different animal populations, and then we screen those samples using molecular technology, sequencing, virus isolation, and we look also into bacterial population in the wild animals, and we check, and also we look into swine populations. Recently we have detected a variant swine virus in swine population in South Africa, and also we conduct human clinical studies. We do zero prevalence on the animal workers in the field. So our hypothesis is that we look into the zoonotic and antimicrobial resistance pathogens at in the livestock production system using a human-animal environmental approaches, and also we look into the distribution of those viruses. We conduct these studies in a population which we haven't been looked before in, for example, a country like South Africa. And my group, we use advanced molecular technologies using serology and real-time PCR, virus isolation, and also we use high throughput sequencing to detect novel viruses. And for example, taking influenza virus, as you know, influenza is available in different hosts, and we have now 4 subtypes, A, B, C, and D. And from these nice wild birds, we are being challenged with new viruses. And here we use a One Health approach to tackle pandemic influenza virus in South Africa, where we collect samples from animals and humans on the farm. And actually we have detected 5 samples containing H1N1 and 1 sample containing a variant H3N1. And also we collecting the serum from the participants, and we have found those antibodies in their sera. So this reflects that there is a dynamic movement of those viruses between the animals and the humans. And this is the One Health approach that we use to tackle these influenza viruses in South Africa. This study has been submitted now, where, as I told, we have detected H1N1 and H3N2. Another study, we look into the detection of influenza viruses. We have collected almost 1,000 samples from different wild bird populations, and some of these has been published already. Last year, we have detected for the first time in wild waterfowl in South Africa, we detected influenza viruses of the low pass. And this year, this month, actually, we have detected for the first time the detection of H5 and other influenza viruses in migratory falcons, migratory armor falcons, and this showing you the migratory route of these falcons. They migrate from northern China down to South Africa, and actually they cross the Indian Peninsula, the Arabian Peninsula, and we have tracked these using transmitters, and we detected influenza viruses in those. So this also movement of the viruses from one corner of the world into another corner or another part, this is very problematic issue, and we have to tackle this. And also we have detected avian flu in the waterfowl poultry interface in wetlands in Southeast Asia, and also this has been published. Just highlighting about the AMR, also using One Health, we have looking into MRSA and Salmonella enterica and other pathogens of human interest that cause human disease in those animal populations. And most of these studies also has been published. And we also have detected some novel strains containing mutations and such as maize EC8 and 10 and Salmonella and Listeria and other pathogens from those animals. We do whole genome sequence. We found different mutations that may account for antimicrobial resistance. We're still studying those mutations in those strains. This also show you that we have a huge future plan, and all groups should focus on the One Health approach and looking from a One Health perspective to tackle those diseases. Currently, we are looking into mixed infections of COVID samples and also zoonotic AMR, and we will continue these virus threats. My take-home message is, today we should and we have to consider an outbreak somewhere is an outbreak everywhere. We have to work on a holistic approach, a One Health approach to tackle those diseases and those threats. And still, we can imagine this scenario. You are on a plane and then they are coming, the surveillance team, to investigate the threat that you are bringing into a new country. And this image is very expressing one showing in 1918 the influenza pandemic, and here's the 2019 COVID pandemic, and we still suffering from this pandemic. We don't know yet when it will end, hopefully with the vaccine and the antiviral scale up. And again, we have to be well prepared for those pandemics because we are same what we have prepared for influenza, we have to be prepared for this COVID pandemic and other pandemics. And I have to thank my grant support funders. We are supported from the NIH and the Center of Excellence for Influenza Surveillance. And without this collaboration with different universities and different institutions, this research wouldn't have come to light. And acknowledgement to Erasmus Medical Center, South Africa National Foundation, Duke One Health, and the University of Minnesota, Uppsala University. All these support our research, and I would like to thank you for this opportunity and for this meeting. Thank you so much.
Thank you very much. And Dr. Jitendra, are you ready with the questions?
Just I'm screening some of the questions. We have received the very large number of questions. So there's one participant is asking about how vaccination and One Health play a mechanism in the vaccination.
Can you please repeat the question, please?
The one participant has asked about the how far the vaccination and One Health mechanism play a role in it.
You mean the vaccine?
Yeah.
Play a role in what?
The One Health.
Oh, I see. Vaccine is a prophylactic measure that will prevent the disease, but One Health is an approach. Yes, vaccination are important, but One Health means that we have to look into the virus emergence or the pathogen emergence before they occur. And that's why we use the One Health approach to detect the viruses or the pathogens before they reach the community. Like now, we are still struggling to find which effective and efficient and safe vaccine for the COVID-19. So we use the One Health approach to prevent those pandemics to jump into the human population.
Okay, sir. So, Dr. Santanu, sir, do you have any questions?
Yeah, there is one participant is asking one question with the permission of Dr. Jitendra. I'll take it. It's an interesting question by the participant. Professor Zolati, Professor Mays, this is a question for you.
Yes.
Since global warming or temperature is increasing day by day, and this is leading to melting of ice or glaciers, and this is making us more exposed to various giant viruses which have hundreds and thousands of protein synthesizing genes. And while HIV, one of the deadliest viruses, just have 9 genes within it, and if such virus unfortunately attacks the mankind or humankind, what are the best possible approaches that we should adopt in current time so that we should be able to deal such a situation in future? So what would be your response? We are
Yes, I am aware of this giant virus. It has been detected probably since 2003, and probably they were there. And yes, I agree with attendee who asked the question that global warming gets a factor. If you remember one of my slides showing to you how the environmental factors are affecting infectious diseases, definitely pollution, global warming, and other environmental factors will ignite some of those pathogens to jump to other hosts. However, so far, there has been no human infection with those giant viruses. These viruses infect amoeba and other protozoa, as you may know. But so far, luckily, there has been no infection with those giant viruses. But we don't know what will happen in the future. And as I said, yes, this is also under One Health. We look into those pathogens in their original source. And then we detect them and we predict them before they jump, and we study them. And this is the holistic mission of One Health approach. We look into those pathogens in their original source. But so far, I would like to make the person who asked the question that there has been no infection yet with those giant viruses. They infect other hosts, amoeba and other protozoa.
Thank you very much. And over to Dr. Jitendra and Dr. Sethi, sir.
Dr. Jitendra, do you have any more questions?
No, sir. The most of the questions we have received is generalized questions, so we can further proceed.
You are skipping them. Thank you very much. So the outbreak anywhere is outbreak everywhere. Thank you very much, sir. The organizing committee is thankful to you for discussing genetic pathogen, spread of virus, its timeline, and recent data on the emergence of this pandemic threat. You also highlighted the interconnection of SARS-CoV-2 with emergence of various infectious diseases. I firmly believe that the participant must have enjoyed your interesting and interactive presentation on holistic, transdisciplinary, and multidisciplinary One Health approach to mitigate future emerging pandemic threats. Once again, sir, Indian Virological Society as well as the College of Animal Biotechnology appreciate you, sir, for accepting our invitation. Thank you so much.
Thank you, Dr. Sita, and please again allow me to thank all the attendees who attend my presentation, Professor Yashpal Singh, and it's a pleasure to be among with you, working with you. And again, thank you. We will enjoy this webinar, and we want more webinars similar on the same issue. Thank you so much.
Thank you very much, Professor Mays.
Thank you, Dr. Singh.
So our next speaker is Professor Dr. Shalinder Kumar Saxena. Dr. Shailendra is the Vice Dean and Professor at King George Medical University, Lucknow. He is the author of several popular books in the field of medical microbiology, virology, infectious diseases, and nanomedicines. His primary research interests are to understand the molecular mechanism of host defense during human viral infections and to develop new predictive, preventive, and therapeutic strategies for them using emerging and re-emerging viruses as a model via stem cells and cell culture technologies. His talk is on the pandemic coronavirus disease 2019, COVID-19, the unseen and unsaid. Due to time constraint, Dr. Saxena is not presently live and he has sent us a recorded video and we will be playing his recorded lecture. Dr. Jitendra, can you do that?
It's fine now.
Yeah.
Is it coming, sir?
Yeah, it's coming.
Yeah, it's fine.
Hello, greetings of the day. Today I'm presenting some of our work which we are doing with this coronavirus as this has emerged last year. The title of the today's presentation is Pandemic Coronavirus Disease 2019, the Unseen and Unsaid. We have already seen a lot of this, and let's start directly. One of the very favorite slides which I always talk about is my name is Khan. And that simply tells that what is the importance of the name. And if you think of this, when this virus emerged, we have given a lot of names. Wuhan virus, novel coronavirus, COVID-19 virus, pandemic coronavirus, and so on. But the International Committee on Taxonomy of Virus, ICTV, has formally given its name as SARS-CoV-2, which means Severe Acute Respiratory Syndrome Coronavirus 2. And this was based on the phylogeny, taxonomy established practices and formally recognizes this virus as a sister to the previous SARS-CoV that we have seen in 2003. Now, how the virus looks like. A lot of images are now available on the various social networking platforms as well as on Google. But these are some of the very original pictures where you can see these are the scanning electron microscope, and the virus looks like very similar to the rays of the sun, and that's like a crown. And that's why it is known as coronavirus. And you can see here, these are the lymphocytes, and the virus is getting mature, and with the help of egress, they are getting out. This is the three-dimensional structure of the virus, how it looks like. And on the surface, we have the spike glycoprotein, which at that time nobody was knowing is that important, but we have shown a lot of studies with this protein. Eventually, at that time, further there is an M protein, there is a hemagglutinin esterases envelope, RNA and N protein, and then followed by the E protein. And now the SARS-related coronaviruses are the viruses from the subgenus Cervicovirus, which is basically this belongs to beta coronavirus genus. And as we know, the coronaviridae family has three different types of the genus, that is alpha, beta, and gamma. So this is under the beta coronavirus. And these viruses are enveloped, usually rounded, but can be filamentous also, composed of a protein, a studded protein, and surrounds the genome. They have been very well known in animals from 1930 onwards. And now very recently, we are seeing from 1960s onwards in the human beings as well. So this was the very unique picture which we have drawn in a very early stage last year, where we have shown that how the virus looks like. So this was a three-dimensional structure which we have done with the help of in silico modeling at that time. And this was the membrane glycoprotein. The nucleocapsid protein was inside, and then there was an envelope here, which was nucleocapsid was having a very specific RNA inside,
And then there was the spike glycoprotein, which has been very important. Now we know the genome is around 26 to 32 kb of this virus, comprising of around 6 to 11 open reading frame. And if you can see, the ORF1 is one of the biggest one, which comprises around 67% of the genome, and it encodes around 16 non-structural proteins called NSPs, whereas the remaining ORFs encode for accessory and structural proteins. The NSPs includes two viral cysteine proteases. It includes papain-like proteases NSP3, as well as the chymotrypsin-like, 3C-like, or main protease NSP3, RNA-dependent RNA polymerases, NSP12, helicases NSP13, and other likely to be involved in the transcription and replication of the SARS-CoV-2 virus. In addition to the NSPs, the genome encodes for the four major structural proteins, which are the spike, matrix, subglycoprotein, membrane and nucleocapsid protein, and envelope and accessory protein like ORFs. So that's how the genome is made up of this virus. Because the genome is segmented, there is a chances of recombination, and that's why there are the various strains of the coronavirus is known, called A, B, and C. A is one of the oldest strain of this coronavirus, which is found in bats and pangolins, considered the root of the outbreak. And the two subclusters are now, one is linked with the Wuhan and the common in America and Australia, and the other is in the other parts. So the B is the type B virus, which is the variation of the coronavirus most common in Wuhan. It is derived from the type A via two mutations and mutates slowly in China but rapidly outside China. There's another strain called C virus, and that is the daughter of type B. It is just one mutation different to the parent variation, and it spreads to Europe via Singapore. So these are the three subtypes or the strains which we know, and A to A is one of the most commonly found in India. Now, how the virus has evolved? If you know, there have been very potential animal origins of the human coronaviruses have been suggested. As you see here, in 2002 and 3, we had the SARS-CoV that was found in the bats and the pangolins, and then we have the MERS-CoV in 2012 and 13, where the camels have been thought to be the intermediate host, and the bats were like, again, reservoir. Now, for this time, the reservoir is believed to be the bats, but a lot of intermediate hosts have been suggested play a very important role, and we still do not know which one is exactly which particular infection animal has played a very important role. So, in all of them, the human-to-human transmission is also reported. So, very important for us to understand this. Now, when we have done the phylogenetic analysis, we have found that this Wuhan seafood market, that the COVID-19 virus was very oftenly found in bat, as well as it was also seen in the civets and the other groups of that family. But it was separate from the JEV, which was a flavivirus, suggesting that it's an outgroup. So the study is really correct. What we have found when we have compared the 2003 virus and 2019 SARS-CoV-2 virus, that it was around 87% same with the previous virus, and there was a significant difference in the receptor binding domain here. So initially at that moment, we were not knowing that whether this has any role, but now we all know that this particular difference, the 13% variation which we are seeing from the previous virus and the new virus, is the actual difference which was making the difference in the pathogenicity, antigenicity, and now has a very important role in the vaccine program. And we have published this work in January last year in the journal called Virus Disease. So we have studied the novel glycosylation sites. The glycosylation means the attachment of the sugar moiety onto the cell membrane. And what we have found that this membrane, when this was attached, plays a very important role in glycoepitopes by the monoclonal antibody recognition, viral surface envelope glycosylation, and it plays a very important role in lectins on host cell surface. And this has a role in a response favoring the host antigen presentation and response favoring the viral attachment and entry in the host cell surface. So when we have looked, we found there were the various novel sites, and that has been confirmed by the various databases that they are novel in the previous virus and the new virus. And we have, so you can see this is the new virus, COVID-19, SARS-CoV-2, and this is the SARS-CoV, that is the 2003 virus. So you can see and you can match, and this whole paper is available now. So that tells the beauty of the sequencing analysis, suggesting at very early stage that these epitopes are actually making the difference at the level of glycosylation. That means the attachment of the virus with the host cell membrane. We have looked for the antigenic sites, and you can see here there was a difference in the antigenic sites. Also, there were very few antigenic sites were actually similar in the SARS-CoV and the novel SARS-CoV-2. At that time, it was known as 2019 nCoV. So based on this, we have suggested a list of epitopes that could be used for the vaccination, and several of them have now been used in the two vaccines which we are knowing. So this was the first structure of the spike glycoprotein, and you can see here this is the overlapping of these two SARS-CoV and SARS-CoV-2 spike glycoprotein. And what we have found that the structure was at 1.39 angstrom, which is still one of the best structure than what we have in the crystal, that is around 3.6 angstrom. And this was showing that there was not significant difference in the structural level, suggesting that the virus was not significantly different in the appearance, but it was significantly different in antigenicity and glycosylation, and that might be the reason for the pandemic. And that's what we have proposed, that because the difference in the glycosylation and antigenicity, there was chance that it can become pandemic, and this we have suggested in January last year, whereas the WHO has said pandemic on 10th March. We have also given a couple of those cytotoxic T lymphocyte epitopes, which can be used for the peptide-based vaccine, and several of them are now already in use. We have also suggested that the use of corona-specific attachment inhibitor can be the choice of therapy at that moment, and based on this, the hydroxychloroquine and several others came for the treatment. So if you see the epidemiology, this is just on January 19th, there were 96.17 million total cases throughout the world, around 2.06 million deaths. Whereas in India, if you see 10.63 million total cases and around 153,000 total confirmed deaths have been reported, suggesting that we are still, we have to watch, and you see the graph has been significantly gone up. In India, the good thing was that the deaths were just 2.55%, and most of the cases, around 63% cases, were discharged, and the active cases were just like 30 to 35%, and this has gone down now. In case age-wise, if you see, most of the cases were in the age group of 20 to 49, and maybe because they were not following the standard precautions which have been suggested. The males were having the higher rate of prevalence. The reason was again expected to be that they are outside and they're not following the mask and washing their hands with soap and water and so on. How the COVID is transmitted, by now everybody knows that it is transmitted via droplets, and the droplets goes and lands to your nose, mouth, possibly lungs, and if it is 1 to 2 meter or less, this can also go to the various physical objects, like you can see on these surfaces, and once somebody is positive, it can transmit to the whole society. So the transmission requires large particle respiratory droplet, which should be less than 6 feet. Contaminated surfaces are another possible source of transmission, and all respiratory secretion body fluids, including the urinal and the stool sample, should also be treated as the infectious material. These are some of the common sources from where the infection can proceed further. The children are a little more infectious because they have one day before the symptoms develop, they can actually transmit the disease to others. And if you see the pathogenesis, when somebody sneezes and coughs, and if there is a virus in the droplet, it goes and lands to your trachea, goes to the bronchus and bronchioles, and eventually settles into alveolar macrophages. And then it starts producing very less amount of mucus, and with the help of this goes and then gets out from alveolar to bronchioles to bronchus. And when somebody sneezes again, you get the same virus, and then it goes to the infection to the new person. And as soon as the new person gets the infection, the cellular immune response get activated, which activates the cytotoxic T cells, and they eventually kill the virus with the help of effector cell as well as the cytokines. And the killing of the infected cell actually helps in giving the signal to the B cell so that they can actually have an antibody secreting plasma cell. And that's how the antibodies are made. And they keep a memory at the later stage so that there is no further infection at the later events when it can happen, or you might get exposure with the SARS-CoV-2. Now, if you look onto the symptoms, that most of the cases, 80% were having very mild sickness. There were moderate cases which had the fever, fatigue, dry cough, and there were aches and pains, runny nose, sore throat, shortness of breath, and diarrhea. And in very critical cases, they have severe pneumonia or a multiple organ failure, and that can lead to death. And this is the case where somebody has some kind of morbidity in advance. This was a very specific day-wise presentation that is available in this presentation. So if somebody is interested, they can see that how on a different days that what are the symptoms happens, specifically how the shock and ARDS and respiratory distress and multi-organ failure happens, and what kind of therapy should be given in that particular time. So this is basically for the clinicians who are really interested to see this in a much more detail. In children, what are the emergency signs and when you should go to the hospital? There is a breathlessness, there's a cyanosis, that means the skin colors become blue, not drinking enough fluids, not waking up or not interacting, being so irritable, flu-like symptoms improve and fever with rash. At the same time, in adults, there might be a difficulty in breathing, shortness of breath, pain, sudden dizziness, confusion, and severe and persistent vomiting might happen. These are a couple of complications which are very important when somebody has to be more careful if they have something like this, like sinusitis, lower respiratory tract diseases, cardiac issues, musculoskeletal and neurological toxic shock syndrome, or secondary bacterial pneumonia with or without sepsis. At the same time, these are some groups which are at the high risk, like children less than 5 years of age, senior citizens more than the age of 65, the people who are taking long-term aspirin therapy, they have a high chances of getting the Ray's syndrome, pregnant women, adults and children with the chronic diseases of any of these things, adults and children who have immunosuppressed like HIV, HBV, and SCV, and the residents and the healthcare workers are also at the high risk. How we diagnose, we all know there have been various case definitions given by the WHO. So based on that, we have suspected case, probable case, and the confirmed case. As we know, the suspected case is any exposure in the for last 14 days. Now we are saying 7 days with a person who has been known with the COVID-19. The probable case is a suspected case for whom the testing for COVID-19 is either inconclusive or for whom the testing could not be performed because of any reason. And the confirmed case is when somebody's RT-PCR is positive, irrespective of the clinical sign and symptoms, that person will be treated as positive. And these are the couple of things which we collect or use for collection of these patient sample. What we collect the nasal swab, throat swab, and then immediately transfer into the virus transmission media. We also collect the lower respiratory aspirate in the intubated patients, and we might collect the posterior pharyngeal swab, nasal washes, and acute and convalescent serum for the further antibody analysis. Is there any treatment or antiviral agents? As we know, there's no specific cure yet available. However, the worldwide people are trying to do a lot of research so that they can find out appropriate vaccines and drugs. So at this moment, the clinical management involves the infection control, isolate the patient, implement infection control and precaution, PPE for healthcare workers and family members. Supportive care involves pulmonary administration of oxygen, mechanical ventilation for respiratory failure, and treatment requires a lot of medication. There's a list which I have shown earlier, and corticosteroid treatment is normally not recommended unless or until there's a very, very severe case. Other than this, we use IV fluids, parenteral nutrition, oxygen therapy, antibiotics for secondary infection, vasopressor for shock. Paracetamol is prescribed for fever, myalgia, and headache. Patient is advised to drink plenty of fluids so that the virus is diluted. Avoid smoking. For sore throat, the short course of topical decongestant or saline nasal drops or throat lozenges or steam inhalation may be beneficial. Salicylate or aspirin is not suggested or contraindicated. Monitoring of the clinical and radiological evidence of lower respiratory tract infection and for hypoxia is also very important for us. So what is the prevention? We all know the prevention is washing your hands with soap and water, maintaining at least 1 meter distance between yourself and anyone who is coughing or sneezing, avoid touching eyes, nose and mouth, stay home if you feel unwell, follow the direction of your local health authorities, stay informed on at least developments for the COVID-19, keep up to date on the latest COVID-19 hotspots in your city. And as we know, now we have at least 4 types of vaccine worldwide. There can be the whole virus, there can be the nucleic acid, viral vectors, and protein-based. And based on this, now there are the 2 vaccines, the Covishield, which is a viral vector vaccine, and the Covaxin, which is a virus, inactivated virus vaccine. So these 2 have been given the emergency approval in India, and both of them have the efficacy of 90%. Now, there are various clinical trials are going on with the preclinical candidates where a lot of vaccines and treatments are in the pipeline, which might come by the end of this year. So still what we are suggesting that you be smart, that means you stay home when sick, use the mask, avoid large groups, refresh indoor air, and 10 feet is better than 6. So that is very, very important in spite of that if you are vaccinated. So what we can do, there are 4 P's which we always suggest. That is, one is the plan. So take enough time to plan who is in your family and community is the vulnerable population, whether you have younger kids or senior citizens at your home. Schools and university childcare arrangements are available, or work, business and government closure arrangements are available if somebody is sick. Prepare yourself for caring for self and family and community, whether there's enough medication and health supplies are available for you, because when you are not well, you don't feel to go there to collect all these things. School closing arrangements are in place. Alternative childcare arrangements are in place because you do not want to make your kid getting sick when you're putting them in any of these common facilities. And whether you have a non-perishable foods and water available with you so that when you are not well, still you can get the supplies of all these things immediately. Vaccinate yourself for the prevention once the vaccine is available. Cover your cough. You can see how the kids are doing it. If you have any chronic sickness, then prevent and manage chronic diseases. And washing of your hands with soap and water and the alcohol-based hand sanitizer is very, very important. And protect. You should know what are the symptoms, when you should go to the doctor, know when to see the doctor, protect the vulnerable population, and breastfeed your baby even if you are not well. And these are some of the emergency numbers which you can use in case of any emergency. 1075, you can use the WhatsApp, 90131 and this is your hand if you're not washing it with soap and water, even if you're vaccinated. So we are talking about the human infection. We have recently seen other animals where these infections have been reported. So are we ready for another pandemic? Still we are not. And thank you very much for giving me this opportunity to speak with you. And this is one of my book, which is the first book on the COVID-19 published by the Springer Nature. You can get it for free online, although the book is also available on the Amazon if you wish to get the hard copy. Thank you very much, and I hope you enjoyed the whole conference very well. Thank you.
Dr. Jitendra, do you have some questions? And since it was a pre-recorder lecture, so we'll take certain questions and we will send it to Dr. Saxena, and the moment he will reply, we will send to the appropriate person. The organizing committee is thankful to Dr. Saxena for sending his pre-recorded presentation. This is again highly commendable since he was not available, but he kept his promise and sent us pre-recorded video. Thank you very much, sir. The presentation, which was aimed in elaborating the molecular structural aspect of this virus, clinical signs and symptoms, and clinical management precautions related with COVID-19. He also highlighted and defined certain epitopes which are being exploited for the development of vaccines. We are very much thankful to Dr. Saxena for his efforts. Thank you very much, sir. And our next speaker is Dr. Laura. And I think before we start, since it's a long time at a stretch, can we have if Dr. Malik permit us 10 minutes tea break, sir? Virtual tea, what we say?
Yes, we can. Just for
Sir, it will help us to stretch our legs. And
I do agree. We are sitting since I think 8 o'clock morning.
yes.
Yes.
So what we can do is we will not log off. We will keep it as it is, but quickly we can have 10-minute break.
11:45. Yes.
11:45. Right, sir.
It
Thank
may
you
be
very much.
11 minutes. Oh, yes. Please.
Okay.
Thank you.
Laura is about to join.
Laura joined.
Yes,
Laura has
she has
joined.
joined.
Ask her to relax and keep her slides ready. We will be back in 7 minutes.
Okay, sir. Thank you very much.
Okay.
Thank you, sir.
Hi, Laura. Laura, can you hear me?
Yes, yes, I can hear you.
So the plan is like we just started. You know, we started at 8 a.m. Indian time, and it's all since last 3.5 hours. It was presentations, and we have given a 7-8 minute quick break. So meanwhile, you can arrange your slides, and we will be ready at 11:45 at 7 minutes after. Is it fine with you?
Yes, that's no problem at all.
Okay, thank you. Welcome back. Hope all the team members are ready. Okay.
Yes, now we can proceed.
Sure. So our next speaker is Dr. Laura McFlynn-Berry. Laura did her Bachelor of Veterinary Science from University of Melbourne. Then she did Bachelor of Science in Economics from University of London and a Master of Science in Veterinary Epidemiology and Public Health from University of London. She is US-based board certified in veterinary preventive medicine and a member of the Australian and New Zealand College of Veterinary Scientists in veterinary epidemiology. She began her career in the state of Victoria as a chief veterinary officer and chief veterinary officers unit as a trainee epidemiologist, moving to the senior epidemiologist for the state in 2013. She briefly joined the Australian government in 2015 before rejoining the department in 2018. Laura has also worked with FO of the United Nations in Bangladesh, Vietnam, Myanmar on animal surveillance projects and as a consultant for the University of Sydney and the state of New South Wales in international epidemiology capacity building. She was second to the Australian Department of Health's COVID-19 National Incident Room from April 20 to work in national surveillance planning. She has been awarded 2019 Australian Churchill Fellowship for an international study tour to investigate veterinary field epidemiology training program, which she hopes to complete this year. She will be delivering a talk on Australian veterinary epidemiologist in the national response to the COVID-19 pandemic. Laura, please.
Thank you, Dr. Sethi, very much for that introduction. I'm really happy to be with you all today and to share the experiences that I had working in the COVID-19 response, as well as the colleagues that I have at Department of Agriculture, Water and the Environment. Let me just see if I can share my screen. Give me one moment. And hopefully everybody can see that up on screen
Yes.
at the moment.
Yes, you
Wonderful.
Go ahead.
So as per the introduction, I'm a veterinary epidemiologist, and I work for the Australian National Government in the Epidemiology and One Health section. And today I'm going to be speaking about the experience during the COVID-19 response that I had, that my colleagues and I had. Just as an overview, I thought I would provide a little bit of a description about our role within the government and also the Australian animal health system, describe the Australian COVID-19 situation, and then talk about our experiences, lessons, and ideas. Just quickly, epidemiology is the study of the distribution and determinants of health-related events or states in specified populations and the application of this study to control of problems. So I'm aware many people listening today may have an idea of epidemiologists as like biostatisticians, I think the more academic side of epidemiology. But there is a whole other area of epidemiology that is about the application. And that's what we practice within a government setting. And so we really look at controlling disease outbreaks and controlling endemic diseases, looking for undertaking surveillance, so understanding what the disease situation is within a region or within a country. And we use that information to inform changes to legislation or changes to policy, different disease control strategies, and other activities which will improve animal health or public health, farm profitability, or trade. So this is where we fit in. It's a very small organization chart, so I don't think you'll be able to see everything there. But my colleagues who I'm speaking on behalf of today, Rachel Iglesias, Sam Hamilton, Andrew Breed, and myself, are located within the Biosecurity Animal Division of the National Department. And one other colleague is located in an entirely different area. He deals with more of the analytics and the biosecurity systems. So most of us who were transferred temporarily across during this response normally work as epidemiologists, but we came to government from a variety of different pathways. So Rachel initially worked in a private practice before she moved to the National Department about 10 years ago, and she has a background in conservation medicine. I think Andrew Breed has also spent a little bit of time in practice and completed a PhD in Hendra and Nipah viruses before he worked in the United Kingdom for some time in a laboratory, and then came back to Australia. So he comes from quite a different pathway. Sam worked in private practice before joining the national government about 15 years ago, and he's completed a PhD in design and analysis of epidemiological models. Greg, on the other hand, started as a field veterinarian working for the government at a state government level in Australia, and completed a PhD in mathematical modelling, and he has worked in data science as well as epidemiology. And as you would have heard from the introduction, my background is at a state government level, but I've also worked internationally on animal surveillance with Food and Agricultural Organization in a few countries in Asia. So we all come from quite different backgrounds, and this meant that we had different things to offer for the public health response to COVID-19. During our regular jobs, though, we were involved in different activities. We help coordinate animal health surveillance across Australia, and we provide technical advice to support trade of animals and animal products and improve the policy situation with regards to both diseases that occur in Australia and those that are exotic. So that's a little bit about us. And in terms of the Australian animal health system, Australia is a federation. And so we have state governments, and we have the federal government as well, or the national government. In Australia, at a state government level, the state and territory governments are responsible for animal health within their borders. So any disease outbreaks that occur, disease control measures that are implemented, outbreak investigation, those are all done at a state level in Australia. The Commonwealth government is responsible for quarantine and for international animal health matters. So we really deal with things that occur at the Australian border, but also perhaps some activities that occur. We support our neighbours to conduct some activities to look for diseases in neighbouring regions to Australia. And we also have a coordination role at a national level. So while we don't implement the activities directly, we support the states and territories to respond to disease outbreaks. And I think this is quite similar to the public health system in Australia. So in Australia for COVID-19, the state and territory health departments were responsible for responding to the disease outbreak. And at a national level, we were responsible for coordination and support. And so this meant that for us coming across from animal health to public health, we already had not only some idea of what epidemiology is and how we could potentially apply it to public health, but also we had an idea of the organization that we were going to be working in and how that would work in an emergency situation, and how we would communicate information to different people within that emergency situation. And as an applied epidemiologist, communication is as important as the analytical side of things. So that was really, really valuable. I think I should also point out some differences as well between public and animal health. With public health, the focus is really on protecting human life at all costs. Whereas I think in animal health, we have a slightly broader focus. So we're looking, we do a lot of preparedness work to help us have the skills to respond to disease outbreaks when they occur, but also to demonstrate to our trading partners that we're capable of detecting diseases such as foot and mouth disease, which are exotic to Australia. And that helps us support trade of animals and animal products. We also undertake a lot of risk assessment and regulation at the border. And we also look at biosecurity at an enterprise level. So making sure that our farmers are able to implement measures on farm which will protect them from disease. And we really consider biosecurity a shared responsibility that governments and industry and farmers share together. And that's a little bit different to public health. But despite these differences, I think we were able to work quite effectively within the COVID-19 response in Australia. I thought I would just quickly mention the situation in Australia because it is quite different to the situation in other countries at the moment. So Australia experienced 2 large waves of COVID-19. The first really associated with people returning from overseas, and that happened sort of March to April last year. We had a second large wave that just occurred in the state of Victoria, and that was associated with quarantine, a release of virus from quarantine. So in Australia, from sort of the middle of last year or earlier, we have required anybody entering Australia to, there is a mandatory hotel quarantine program. So you have to spend 2 weeks in a hotel quarantine that you pay for yourself. And we've also stopped people who are not Australian citizens or permanent residents returning to Australia. So the number of people entering Australia right now is really greatly reduced, and that has minimized our risk a lot compared to some other countries that cannot control their border like we can. There's really some advantages to being an island. We also have actually stopped Australian citizens and permanent residents leaving Australia as well. And so you actually require permission to leave the country at the moment. So this measure, and I think we have quite a strong public health service in the states and territories, has enabled Australia to really effectively suppress COVID-19. We are having very few cases, if any, of community transmission at the moment. So the cases that are occurring are associated with international travelers coming in, and they're located within hotels within that quarantine program. So I think these strict border controls and the strong public health service have really enabled Australia to suppress COVID-19 in a way that many other countries unfortunately just can't. And so certainly in Europe and in the US and in India, I think the situation is very, very different. Here are some key points during the response. So you'll see that in March, all overseas arrivals were required to self-quarantine for 14 days, and that happened at home. We found that some people were not adhering to those instructions, so that mandatory hotel quarantine started right at the end of March in early April. And then we did see, again, as I mentioned before, that surge in cases, and that was associated with a couple of releases of virus from quarantine where infection control procedures broke down. That's not only happened in Victoria, but in other states and as well in Australia, but more fortunately, those outbreaks have been contained very quickly. And so I just thought I would quickly mention the public health system in Australia. So when I talked before about animal health, public health is very similar. The states and territories are responsible for all of the activities within their state or territory. And at a national level, we really help to coordinate what is happening and make sure that as much as possible the activities are harmonized. We also help support outbreaks when they occur across multiple states and territories, and help support resource allocation as well. So making sure that enough masks and personal protective equipment are available in each of the states or territories. And that leads me to talk about our experience during the COVID-19 response. So there were 5 of us that were temporarily transferred across to different departments. In orange on the bottom of your screen, you'll see that 4 of us went across to the Department of Health in the National Incident Room. So that's the emergency control center at a national level for health. Greg Hood, other colleague, went across to the Prime Minister and Cabinet, which is a different department, but he was doing similar activities within that department. The National Incident Room is the centre that coordinates the activities for COVID-19 across Australia. It was activated by Australia's Chief Medical Officer in January 2020, and it's mostly staffed, most of the staff are from with the National Department of Health, but there were also people that were brought in from other agencies, from the university sector, from other departments like myself and like my colleagues, and also people from the Australian Army and Navy and other armed forces were there as well. So Andrew Breed, my colleague, was the first to participate in the response, and he was there from March until July. He was initially involved in reporting against metrics that were required by the leadership to help make decisions during the response, and then was involved in weekly epidemiology reports, and later transitioned to supporting molecular epidemiology or genomics, which we've used really successfully in Australia, and I'll talk about a little bit later. Greg Hood, as I mentioned, went to a different department, but he was also involved in reporting against key metrics and helping design databases and systems for data management. The need for data was really quite critical in this response, and there was an expectation that data would be available in almost real time. I'll talk about that a little bit later as well. For me, I was in the National Incident Room from April until June, and I was involved in developing Australia's national surveillance plan and testing framework, and also participated in committees on looking at sero surveillance, which fortunately we haven't needed to use so much in Australia because the incidence or the number of infections that we've had has remained relatively low, and also the use of wastewater to detect COVID-19 cases. Sam Hamilton was in the National Incident Room in July and August, and he was really involved again in reporting, but also providing advice on conditions needed for Tasman travel, so between New Zealand and Australia. And also at that time, we had a large outbreak in aged care facilities where elderly people live. And so he was involved in developing some of the policies around that. Finally, Rachel, she was actually there from August until December, and she was involved a lot in risk assessments and looking at which countries we could potentially have people come from and enter Australia without quarantining. So that's still a work in progress. At the moment, some people can come from New Zealand to Australia, but we can't go back to New Zealand. And there are no other countries that Australia is accepting people from without quarantine. So really at the moment, Australia has isolated itself off from the rest of the world, but that really has enabled us to control COVID more effectively than I think countries that cannot control the borders so well. And that kind of leads me to some lessons and ideas that we have had through working in the response. It is really the largest response that we have seen in our lifetimes, and there is a lot that we could take from the public health side of things that could help us in animal health better prepare for emergencies, but also better prepare for animal emergencies, but also help us prepare for the next human pandemic, which potentially could happen in our lifetimes. One of the most valuable things that we experienced participating in the public health response was the benefits of cross-disciplinary collaboration. So our skills from the animal health side were transferable across to public health, and we could make a valuable difference to the public health. Well, I like to think so, to the response. It was all very rewarding for us to be able to use our skills to help our public health colleagues in this way. I think it's important that to realize that the public health, epidemiology and veterinary epidemiology are slightly different disciplines with that different focus. And so I think as veterinary epidemiologists, we could bring some different skills and ideas to public health. Now, while all of those ideas weren't necessarily helpful in public health, there were quite a few things that we could help with, and that risk assessment process for Tasman travel is one of them. We do have a lot more experience conducting risk assessments on the animal health side of things. I think it was also an opportunity for us to get an insight into the way that public health epidemiologists and public health practitioners respond to a public health emergency. And we got a lot of ideas about what we could do to prepare better on the animal health side of things. I think having this exchange between people of different disciplines really enables innovation and facilitates information exchange, which hopefully enables us to respond better than we otherwise could to this outbreak. Hopefully these are relationships that will continue after the response to COVID-19. There are a lot of issues that really are One Health issues, and there's huge benefits to collaborating with other sectors. I think one of the biggest things we took away from participating in this response was the information needs that are required by decision makers and by politicians to help them decide the best course of action to contain the public health emergency, but also the wider implications of a public health emergency on the economy, on education, and other sectors. Really, the demand for data was continuous, and the expectation was that we could provide data in real time. And you know, as a federation where the states and territories are responding separately, it can be very difficult to provide all of the different types of information that was needed nationally at an almost real time, And so that really was a challenge that I don't think any of us were quite expecting, how much information was required. And to do that to have that kind of information sharing requires really modern information technology infrastructure and also workflows that enable information to be shared between different organizations all of the time. And so we really required systems that were flexible, that could respond to different needs and provide the information that was required. And that was a little bit challenging at times. Australia's health infrastructure was established, the National Notifiable Disease System was established in 1990. So it was established for rapid but not real-time information exchange. And so we really needed to think of different ways to enable that information to be transferred between different organizations. I think it also really highlighted how much politicians and health decision makers need information to be synthesized into something that they can understand really quickly. So short documents or infographics that will explain the situation without really needing to read through very long documents. And from the animal health side of things, we also have a similar kind of legacy system, which we're in the process of now updating and modernizing to make sure that our systems can provide the level of data and information that will be demanded of us in an animal health emergency. Many of the documents and graphics that I've mentioned today, the Australian government, the national government, but also the state and territory governments have made available online. And I think this is really important because it enables people to rapidly understand the situation and make their own decisions on you know, a range of different things, not only their health, but their business and what they're planning to invest in. I think it builds trust between the government and between the population, the general population, when the government is able to provide a plan and that everybody can read if they want to. I think in the animal health side of things, we do try and make some documents available, but I was certainly impressed by the range of materials that were available on the public health side. As I just mentioned, I think communication, it's not only transparency, but communication that's required to help everybody understand what the situation is and use that information to change their behavior, to design interventions that will improve the health situation. So we've had some interventions designed for aged care facilities to minimize the risk, also at quarantine facilities to minimize the risk that the virus will escape out into the general community. And that information can be used to improve policy as well. So to be communicate effectively is very important. I think, you know, in a similar way as, you know, India has states and a national government, Australia has a similar structure, and that often can be very, very challenging. We all have a role to play, and having some of these coordination mechanisms at a national level enabled the best practices of one state to be shared rapidly with another state or territory that perhaps didn't have those same control measures in place. For example, the state of New South Wales had a very effective contact tracing system where they could follow up with the contacts of people who had been infected. And that was a system that Victoria adopted late last year to help them better be able to respond to cases and outbreaks when they do occur. We also have different mechanisms for the general public to be involved. We encouraged people to present at testing clinics if they had clinical signs. So really encouraged everybody to be tested and to stay at home and isolate if they had any clinical signs to minimize the risk of transmission to other people. And I think that's a luxury Australia has as well that doesn't exist in many other countries. We have a website called Flu Tracking where people can sign up voluntarily and each week report what's if they have any flu-like symptoms, and this is tracked over the course of the outbreak. So you can go onto the Flu Tracking website and see how many people had a cough and fever for the past year. So that system was enhanced and built on to really try and involve the community and as another way to detect cases if they were occurring. I think I've alluded to this a few times, but the importance of having mechanisms to enable data and information to be shared between all of the different organizations in Australia. So one really good example is the Communicable Disease Network of Australia. And that usually meets fortnightly to help exchange information on different diseases, infectious diseases and communicable diseases that are occurring, but has been meeting more frequently during this response. And they also developed something called the Series of National Guidelines, which are very specific instructions on how to respond when an outbreak occurs and how to respond in different kinds of settings like elderly aged care settings, with people with disabilities, for cruise ships and so forth. So I thought that way of developing how Australia will respond, and those are again available on the website and anybody can download them, was really beneficial. I think I've mentioned surveillance a few times, but for those who don't come from an epidemiology background, surveillance is the systematic and ongoing collection, collation, analysis of health-related events and the timely dissemination to people who need that information to action it appropriately. Again, one of the strengths of Australia is that we have a number of systems already established for influenza-like illness. And so for COVID-19, those systems were enhanced to more easily. So we really built on what was already there rather than developing whole systems from scratch. I think that was very beneficial to have something already existing. And I mentioned at the start that my colleague Andrew Breed was involved in the genomics side of things. In Australia, we've been sequencing over 50% of the cases, and that sequencing information has really enabled us to better respond to outbreaks, to link cases together, and potentially understand how cases have emerged in the community, so where they're linked to quarantine facilities and how that's happened. So genomics, I think this is the first, genomics has been used in the past for other diseases, but this is the first time it's been used at such a large scale to help us detect outbreaks and contact trace and link different cases together. A couple of other aspects from the laboratory was wastewater surveillance. So at different areas of Australia, they're testing regularly for COVID-19 in the wastewater and using that information to warn communities that there could be cases in the area and to go for testing, and if they have clinical signs. I think we've also used some portable testing, particularly in some remote communities where Australian Aboriginal and Torres Strait Islander people may not have ready access to laboratories or other facilities. And so, and also other people that live very far away. Australia is a very big country. And so some people, particularly in the interior of Australia, may be many hours to a healthcare facility. And so that's where we've used some of that portable laboratory technology to help us detect cases. One other thing that I thought was really interesting was something called the Rapid Research Information Forum. And it involved people from all kinds of different organizations. The government would task them with a question, such as how long can SARS COVID-2 last on surfaces, or what are the most promising COVID-19 therapeutics? And some of Australia's best researchers would then develop a 1,500 word response that they wrote in plain language and uploaded again to the website so anybody can read it. And it really enabled some of the most innovative kind of cutting edge ideas and people to be involved in the response and to provide that expertise from the university sector, where us in government could then use that information to help us design the policy. So certainly working in the response, we don't have time to conduct such extensive literature reviews, and it was very beneficial to have access to universities and to researchers in that way. I think this response has also emphasized the importance of preparedness. And an emergency public or animal health crisis can happen at any time, including the next disease X. It is really helpful to have as many people trained as possible and to have those relationships built during peacetime when we're not in an emergency, so that when emergencies happen, people can be brought in quickly and they understand how the systems operate, and they already have relationships, both informal and formal meetings already established with other organizations, because it really helps with data sharing and information sharing, and it's so critical for a rapid response. I also mentioned field epidemiology training. In all of the state and territory governments in Australia and nationally, we had trainee epidemiologists who are completing Australia's field epidemiology training program. And I think this is something, again, we can bring across to animal health. It's a really fantastic way of training epidemiologists, people in applied epidemiology. It's an on-the-job training program, and certainly the public health trainees were incredibly helpful during the response and could really do some more in-depth work to help us strengthen some of those surveillance systems. Just finally, I think emerging and zoonotic infections are going to continue to be a risk, and even more so as we go forward into the future. The pressures that cause the emergence of COVID-19 are only increasing. Our populations continue to increase, livestock populations continue to increase, and land use changes are driving the emergence of disease. And so it's really important that we have relationships not only within our sector with government and universities, but between different sectors, between animal health and public health and the environment. As veterinary epidemiologists, we have a slightly different perspective to disease than our public health colleagues. And so we can suggest new approaches and new ideas that potentially they haven't thought of before, and can also help with skills transfer and mentoring. And that goes both ways. I think in animal health, we can learn a lot from our public health colleagues as well. I think that the more One Health connections that we have, the stronger that we are, and the more capable we are to respond to events like this in the future. So I hope this presentation has provided you some insight of the Australian response. I think it is quite unique, and certainly the situation here is very different to other countries, but potentially perhaps some ideas from Australia may be useful in India as well. And so I thank you very much for your interest and attention today. If you have any questions, please do let me know.
Dr. Bedi, do we have any question?
Yes, sir. We have some questions.
Okay.
First of all, I'm very thankful to Dr. Laura for his wonderful presentation. And we all fascinate towards the epidemic curves and the dot plots and the data related to the epidemiological investigations. One of the question is that Laura, you already told us about the wastewater surveillance. You were part of wastewater surveillance. And did the Australian government has imposed some strategies to control the environmental persistence of this virus? Since we know that it may persist in environment, it can be transferred to other host.
That's a really good question. I think for wastewater in Australia, there are a number of facilities around Australia that are responsible for processing the wastewater, and not only for coronavirus, but also removing other pathogens as well. And so that's not something that we've been particularly concerned with. The surveillance, though, they take samples before it is processed, and that has enabled us to understand better where outbreaks have been occurring. There is some issues interpreting the data sometimes, because people can continue to shed virus for some time after infection, but that virus isn't necessarily viable. There was and there is a strategy for wastewater surveillance, and that is trying to be integrated into the case detection and the public health kind of surveillance. But in terms of environmental contamination, it's not such a big issue here.
Thank you so much. another question is from my side.
So Laura, Dr.
So-
Bedi himself is epidemiologist, so you can expect a few questions from him.
Not you, only one. So Laura, you have shown the epidemiological case which were reported in Australia. So could you please give some information about the rise of second wave in Australia, since Australia have imposed a very stringent guidelines and regulation for the control of COVID-19. But what were the risk factors were identified for the second wave?
Yeah, so that second wave was associated with 2 virus releases from quarantine, where people were meant to be in quarantine and it was meant to be secure, but because of failures in infection control, they managed to infect somebody who was within the Australian community. For this particular outbreak, many of the people who initially worked in the quarantine facilities had many different jobs. And so when they did become infected by somebody who was meant to be quarantining, they went to a different workplace or they interacted with family. It happened, this second wave happened as all of the states and territories in Australia were reducing their control measures. So initially there were very strict control measures. People were requested to work from home and stay at home. Again, this is a luxury that we have in Australia that many other countries do not have. And at the time in June, there were almost no cases. And so they were allowing larger gatherings and people were starting to move a bit more. People were not so afraid of getting infected. Everybody had got over the initial shock. And it was also in the winter time here, so people are more likely to be inside. And we know that transmission is greatly increased when people are in proximity indoors. And so unfortunately, it was a combination of things that caused the outbreak to get so large at the second time, where it was initially introduced into communities that didn't present for testing, that had multiple jobs, that had large families, and where it was a season where people were more likely to be indoors, where potentially it was in an area of Australia where they've identified the contact tracing system was not as good. And so when cases started to occur, they were quickly unable to trace all of the cases. And so then the only way to control the outbreak was to put everybody in a very strict lockdown where they were only allowed outside for 1 hour a day. So eventually the public health system was not good enough and they had to be very, very strict. And that was the only way they got the outbreak under control.
Thanks, Laura, for sharing your views about the second wave. Now I hand over to Dr. Sethi for next level.
So before we start with the next, on behalf of the organizing committee, we are thankful to Dr. Laura McFlynn-Berry for sharing her frontline experiences of COVID-19 situation in Australia, including surveillance, lessons and ideas, and COVID-19 response as a member of Australian Animal Health System. She has not only described COVID-19 from epidemiologist's point of view, but also defined an important role of veterinarian in the national response to COVID-19 to combat and control pandemic situations with new ideas. I personally of firm opinion that interdisciplinary approaches, even with social sciences and communication skills, along with good governance, are always helpful to define and solve any existing or re-emerging problem. Once again, College of Animal Biotechnology and Indian Virological Society appreciate Dr. Lara for accepting this invitation and delivering a thought-provoking talk. Thank you so much.
Thank you very much for having me.
Thank you. So our next speaker is Dr. Himanshu Chauhan. He has already joined, and Dr. Chauhan is Joint Director and Officer in Charge, Integrated Disease Surveillance Program, National Center for Disease Control, India. Dr. Chauhan pursued advanced fellowship at CDC Atlanta and at Maulana Azad Medical College. He is a public health expert with more than 15 years of experience in diverse capacities and situations, ranging from technical, medical, undergraduate, and postgraduate in a med school. He's teaching in a med school to undergraduate and postgraduate students, and leading public health response in emergencies and policy level engagements with diverse stakeholders. He will be speaking on COVID-19, current Indian scenario, and future directions. Dr. Himanshu, please.
Very good afternoon. Thank you very much, Dr. Sethi and Dr. Malik and the entire organizing team. I would, at the outset, like to apologize for the background noise that probably the viewers are listening at the moment. But since I had planned to deliver this talk from the quiet environment of my office room, but suddenly I was deployed to Jodhpur due to some public health emergency. And here I am making best use of the available resources. I'm connected through my laptop, and my mobile is providing the hotspot signal. So there might be some loss of data packets in between. I apologize for that. The topic that's been assigned to me is I'll be talking about COVID-19, the Indian current situation, and where are we heading to? What could be the future scenario like as far as the Indian situation is concerned? The slide controls are with the organizing team, and I'll request them to go to the next slide, please. So we all know, just a recap of the scenario over the last year, 2020, how it emerged from the end of 2019, when there was information on viral pneumonia, new viral pneumonia in Wuhan, to the time that it was confirmed within a span of just 1 week, WHO had confirmed. And by the end of 30th January, it was declared a PHEIC, which is the highest level of emergency to any public health threat that can be accorded by the global health community. The disease was named as COVID-19, which is what we are using currently, the terminology, as early as 11th February. And then it was a pandemic by 11th March, meaning that it had engulfed all the regions of the globe in the first 3 months of the last year. Next, please. These snapshots, the next 4 or 5 slides, they are just an attempt to capture as to how the pandemic unfolded, starting from China. January was the time when it was confined to mainland China. And then by February, there were lots of countries reporting small number of cases, but mostly it was the Southeast Asian countries which were in the immediate vicinity of China, which were affected, with a few countries in Europe also reporting a small number of cases. As we move to next, please, March and April, the theater where this COVID-19 pandemic unfolded, it shifted towards Europe. And while mainland China by March was still the leader in terms of the total number of cases, but then clearly there was a very significant rise, surge, I could say, in the European countries that we were witnessing. By April, we were already seeing US as the leading country. It had established in terms of the total number of cases. But the situation from European countries, especially Italy, was concerning, and the world is witness to the scenes that were televised by media, that hospitals are full, people are dying. So that the terrible picture, and Italy and Iran were two countries from where these two the worst phase of the pandemic, as we have seen, was evident at that time. Next, please. May and June, by that time it was established, the Americas, North and South American countries were the major countries from where cases and deaths were reported. India was there on the scene. It had emerged, and we had started seeing an increasing trend in number of cases. Next, please. And as we moved to July, August, it was US, India, followed by some European countries, South American countries, which were leading the total, driving the pandemic, as they say. And passing on, we could quickly skip through the next few slides. September, October, it was the time when there was a resurgence in Europe. And we'll talk about the UK variant, which could have played this role by November. Americas and Europe, again, UK driving the resurgence because of the UK variant, which had established by that time. And then December, next please. Americas, Asia, Europe, and by January, the last slide in this series shows that this is where we stand. US clearly is an absolute number of cases. It is way ahead of the next country, which is India. So more than double the number of cases that we have, and then Brazil, and then European Union countries. So this is where we stand. If I talk in terms of numbers about the global snapshot, we are looking at 20 million. We are looking at a number which is total number of cases. United States at 24 million, followed by, because India I'll be talking about separately, then followed by Brazil, Russia, Spain, South Africa, France, and UK in that order in terms of the total number of cases. India, on the other hand, yes, in terms of the total number of cases, we are at number 2 in terms of that, but we'll talk about our response in much more detail. So if we look at the growth rate, what's more concerning is not just the total number of cases, but if we look at the yellow dots on the slide, which show that what has been the growth rate. So the growth rate in the top 10 countries, it's hovering around the 5% growth rate mark, with some of the countries going up to 8-9%. The UK currently is at 9.16% growth rate, Spain 8.29%. But India, on the other hand, if you look at India, just below that 1% growth rate, 0.94%. What's clearly in the top 10 countries, that's the outlier as far as the growth rate is concerned. Next, please. Next, please. Yeah, we also look at the day-wise confirmed new cases and the 7-day moving average of the growth rate. What we see is that since September, September was our peak month in terms of number of cases and deaths. And from there onwards, we are definitely seeing a decline, which is very persistent, which is very continued. So from October, November, December, and we are into the 4th month running straight where we are seeing a continuous decline. Next, please. Overall recovery rate, epidemiologically, the recovery rate, epidemiologists don't like to talk about the recovery rate that much because we know ultimately when this pandemic ends, the recovery rate of most of the countries is going to be almost similar, with most people recovering, more than 97% people recovering. So overall, we are at that mark now, from 96.7%, almost 97% is the recovery rate. With few countries, what this tells us is that at different parts of the country have seen different surges at different times. So the recovery rates might be slightly different in the various states. So while Kerala is currently seeing an upsurge, obviously its recovery rate will reach that level in some point in future. Whereas the other states which are now seeing a definite decline, their recovery rates have peaked or are near peaking. Next, please. If we look at the active, this is what I was talking about. September was the month when we had the most number of active cases. And from there onwards, because of both the decline in the number of new cases as well as the recoveries, we are seeing a continuous decline, which is persisting in January. And we hope by February, we should be coming down to a level where we have less than, we already have less than 2 lakh, that's 200,000 active cases. And maybe by February, we should be having, if the trend continues, we should be having less than 100,000 active cases in the entire country. Next, please. This is another slide showing the same information that from 1st of October onwards, that's where you see a continuous decline in this, in the height of these bars, indicating a decrease in the number of active cases. Next, please. In India, as I said, India is a huge country, and coincidentally, the previous speaker, Dr. Laura, was from Australia, which is we epidemiologists and public health specialists in India, we often have mentioned this fact in our classes teaching to UGs and PGs that India adds the equivalent of an Australian population every year to its net population. So, with that kind of a population base, we are looking at many countries together if the disease transmission dynamics are talked about. So while on the one hand we have states like Maharashtra, which have almost their numbers are like if we look at the total numbers, they could beat many countries in the world in terms of total number of cases and deaths. But on the other hand, we have very small territories like Lakshadweep is the most recent one to become infected. Till 1 year almost, we had been able to successfully hold off the infection from Lakshadweep. But then last week, that also happened, and Lakshadweep was the last district to become involved. But we see that there's a huge variation in terms of the growth rate as well as the number of cases. These are the top 12 states in India which are mainly driving the epidemic. Next, please. This is an interesting slide. When we started off, there were many things which led to an increased mortality, and we were probably slightly above the global mortality rate, case fatality rate, as we say. And but since the earliest, maybe mid-June last year, mid to the current timeline, we have seen a continuous decline. And over the past couple of months, it's slightly started to stagnate at around 1.4% level. And although we are very much at the national level striving towards a further decline in the mortality, but 1.4% seems to be a mark where further decline can happen only with extraordinary measures, both in terms of disease prevention as well as some management therapies which might emerge in the near future. Next, please. And yeah, so if we look, we talked about the top 12 states which are driving this epidemic in India, and even within those states, it's not like uniformly spread. So if I say Maharashtra isn't uniformly affected. It might be the number one state in terms of the total number of cases and deaths in India, but it is not uniformly affected. So if we look at confirmed cases, if we take Delhi as a district, the reason why Delhi has been mentioned here in top 20 districts is because we are considering all the 11 districts of Delhi as one district. Otherwise, Delhi is way below in this hierarchy list. So Delhi confirmed cases as one district, number one. But if we look at the active cases, Pune has the maximum number of active cases. And if we look at deaths, then Mumbai happens to be that district which has maximum number. Now, if you look at Mumbai, Mumbai is at number 4 in terms of total confirmed cases as a district. And Mumbai happens to be at number 4 or number 5 in terms of the active cases. But then the deaths, it's more than any other district in the country. So what I mean to say here by this slide is that there are lots of variables which decide. It's not just the total number of cases. It is population density. It is the availability of healthcare facilities. It is the surveillance and the response system which has worked in those districts, which have all resulted in this picture that we are seeing. Next, please. This is again a timeline of cases and recoveries, which goes on to show that since the lockdown period, the first lockdown and then the subsequent unlock periods, the recoveries have been on a continuous increase. And now, as I was saying, that there's a stagnation. Probably we have reached that level of recovery, which is like 90, around the 97, 98% mark is where probably all recoveries. Ultimately, at the end of the pandemic, most countries would be reporting a recovery rate of closer to this figure. Next, please. So what has been India's public health response? If I quickly want to encapsulate in a couple of slides, India has gone from one or two labs in the very initial part of the pandemic last year, January, February. So the first cases which came to India, there was only one lab which could confirm it. That was the National Institute of Virology at Pune. From there, we have now reached to 2,345 laboratories which can confirm, and this is RT-PCR that we are talking about. So there's a huge upsurge in the total number of the lab capacity in the country, and people are getting even when we look at other countries which are resource rich, there's a significant time lag between collection of the sample and availability of the report. And it's with the availability of the report that public health action starts for certainty. So, but in our country, most of the places we see that within 24 hours, the RT-PCR report is available. That's a tremendous achievement in a very short span of time. If we look at the total the summary snapshot I had already shown, but what this slide goes on to show is that a case fatality rate, which is amongst the lowest in the major countries globally, our active caseload is on a continuous declining trend, and very soon we should be able to see the curve hitting the baseline again. Next, please. This is another aspect which has brought us to where we are in terms of the recoveries, in terms of the low case fatality rate, and in terms of handling the pandemic overall in a way which has given us global recognition. So we have dedicated hospitals in each and every part of the country, depending on what kind of case, whether it's a mildly symptomatic, a moderately symptomatic case who might require oxygen, to a very severe case who might require an ICU facility. These capacities have been developed in a very short span of time. At the start of the pandemic, India did not have the numbers that we are seeing currently on the slide. Our total ICU bed, what we are looking at right now, is 80,485, and 40,000 ventilators, more than 40,000 ventilators. This wasn't the case when we were in the initial part of the pandemic. There were hardly handful of ventilators and ICU beds across the length and breadth of country. So this has been done in a very short span, and it is indeed commendable because of the recognition of the problem at the highest level, and then a decisive action being taken in the shortest time period possible. Next, please. So, if I were to summarize the response to COVID-19 in India, timely recognition of the problem is a huge step, positive step that has led us to where we are currently. It could have been worse. We are witness to scenarios from various countries, but we recognize this problem very early on. Restrictive measures, even at the cost of curtailing people's liberties for short durations. There were inconveniences, but thankfully, the general public, by and large, cooperated, and we were able to take that decisive action. Timely sensitization of surveillance and response system was another step. I represent the Integrated Disease Surveillance Program in India as its officer in charge currently. And I'm thankful to all the state and district surveillance units. All the 733 districts in India have district surveillance units. And I'm thankful to each and every one of them that it was their basic mandate. Surveillance and response is their basic mandate. So they were, with the limited resources they had. Now everybody is talking about the disease surveillance program in India. So that came across as a huge asset in responding to this pandemic. Rapid ramping up of the lab capacity. I've already talked about this particular thing, from one lab to more than 2,400 labs. We have clearly demonstrated that with the will at the highest levels, it can be done. Decisive action at the top level. I said this included both the lockdowns that were put in place, restrictions on international flights, mandatorily making people undergo tests at points of entry, then in the community, then having containment zones, restricting the movement of active cases. All these steps were because there was an agreement, unanimous agreement, and a decisive action at the top level. The time bought through lockdown was utilized for strengthening infrastructure and response systems. So lockdown did not simply meant that we are halting everything. It was the time which was very judiciously used in ramping up the lab capacity, as well as the various types of hospital infrastructure that we are currently witnessing. And whole of government approach. It was not just one. Health ministry could never have handled it by itself. None of the governments across the world were able to do it by their own. One single ministry or one single department. So it was the entire whole of government approach which was helpful in dealing with it. Next, please. Still, there are issues and challenges that remain with the foremost issue being that even after 1 year, the virus behaves in certain ways, and which is not very readily understandable, and we can still say that it's a new virus. So there are lots of unknowns in terms of right now everybody is talking about the strains, the variants. So what effect would these variants have? UK is witnessing an upsurge, unprecedented. And could that same thing be happening in other parts of the world if the strain were to establish? That remains unknown at the moment. And then the implication of that strain with the vaccination drives, on the vaccination drive. So would that new strain be resistant to vaccines, or would it be amenable to control by vaccination? That's also an important. Or would there be? I'm not just talking about the UK strain. There, the virus is also in a race to establish itself. It is there to evade the human immune response and the defense of the human system. So it's a struggle for the virus itself. So where does it stop? It is not clear at the moment. The modes of transmission, initially it was thought that it's only a respiratory virus. Predominantly it remains so. But then we have clearly seen there are other modes of transmission, including vertical transmission as well. So the modes of transmission are multiple. The roles of asymptomatic. At the start of the pandemic, it was thought that only the symptomatics could spread. But now the role of mildly symptomatics and even asymptomatics, totally asymptomatics, is firmly established. The multiple clinical presentations from an influenza-like illness, from asymptomatic, I should say, from asymptomatic cases to mild influenza-like illness to severe acute respiratory illness, and not just respiratory, the other systems being affected as well. It could present as GI complications, it could present as neurological manifestations. So multiple clinical presentations are there. Immunity after infection, that has been in limelight, this particular aspect, that once a person gets infected, how likely he or she is to be reinfected. And we have certainly seen confirmed cases of reinfection. So that immunity is not definitely a lifelong immunity. And the vaccine, ultimately, what would be the effectiveness of the vaccine? This is the current topic which is being discussed everywhere. What is the effectiveness of the vaccine? And the vaccine hesitancy which is associated with it. We also have issues in terms of lack of examples of sustained control at the global level. The moment travel restrictions and other public health measures are eased, we have seen that the virus has made a comeback. China is right now witnessing in some provinces an upsurge after so many months. So sustained control, how would we be able to establish that particular? That is an issue. Contact tracing, when we have smaller populations, contact tracing becomes easier. But looking at a country like India, where we have more than a crore cases, and if we were to identify 15 to 20 contacts per case, we are looking practically at 1/5 of the Indian population as potentially exposed and suspects, and therefore need to be put under some sort of observation, surveillance. So huge challenge. And the data that flows out of all this exercise, the management of that data, it is voluminous data, but there are multiple aspects of that data. And where do we store that data? Who manages it? What level access is to be given to different stakeholders? Those are big issues we have tried to address to the best of our capabilities. But then, yes, there's always a scope of improvement. And lastly, effect on non-COVID healthcare services. We have clearly seen that other government priorities in terms of the public health interventions, they have suffered because of the COVID response, which obviously because of reasons which are very well known, that became the predominant consumer of resources. But then slowly we are returning back, and our focus is now on non-COVID healthcare services as well. Next, please. So, what is the way forward from here? Clearly, one of the tasks would be to take the epidemic curve to baseline. We have seen that it is on a continuous decline, and the ultimate endpoint would be to bring it down to that level where there are hardly any new cases, and all the cases have reached their outcome in terms of recoveries, and a minor proportion of that would be reflected in case fatality rate. Increasing testing and tracing at different, because as I said, that India is not like uniformly affected. There are places where still we are seeing an increase in the number of cases. Kerala is one example. So they are looking at increasing the testing and tracing capabilities, and in most other parts of the country we are seeing a decline. So the testing is also commensurate with the declining numbers that we are seeing. Further reduction in mortality, that would always remain a priority. We try to bring it to less than 1%. That has been the government effort, that we bring it to less than 1% with all available resources. Then vaccine availability for all and addressing hesitancy issues. This is the issue right now that the country is facing, and we have seen an encouraging response in most parts of the country to the vaccine launch. But yes, addressing hesitancy, which is at the level of some healthcare workers, some doctors. So addressing their concerns and then ultimately reaching out to the entire population would be the way forward. And lastly, we'll have to strike a balance, because it's been 1 year so far. Clearly, because of COVID, the country has seen the effects on economic sustainability. People have lost livelihoods. People's lives have been affected in multiple ways. So there has to be a balance between disease control, the cost that we are willing to pay for disease control, and what we see in terms of the economic impact of that particular intervention. So a balance, if it can be struck, and we are right now at that phase where more or less it is established. Most of the economic activities have resumed, barring a few, and in the near future we should see that too being fully resumed. So that's about the global, and then mainly about the Indian situation and the current and the future trends that we could be seeing from my side. I'll be willing to take any questions if there are, and I thank the organizers once again.
Dr. Shantanu, do we have any
Uh,
questions?
yeah. You have a number of questions. Thank you very much for providing, the overview of the current scenario of COVID, and no doubt for future directions you highlighted. Thank you very much, sir. Um, Questions, here it goes from one of the participants from ICMR. Uh, so, what is the cause that Maharashtrians have more cases and deaths reported? Is it their immune systems weaker than other state people, or any environmental factor or social factors play a role in this case? What could be the reason?
See,
One of the participants asking.
yes, and that's a good question, tough question, I would say. And coming from ICMR, ICMR is the research organization which has the mandate. What we are seeing is not just in the case of COVID-19. Let me go back, because I don't have an answer to that question readily available as to why Maharashtra is still, when the entire country has gone into single digit deaths. Most of the states now report deaths in single figures, 8, 9, that's been the number. But when I look at Maharashtra, daily morning, I submit the report for updating the ministry website. And I look at Maharashtra figures and I find that it's 50, around that 50 mark. It's been hovering for a very long time. So what is the reason? And it's not coming from everywhere in Maharashtra. It's from Pune, Mumbai, Thane, and basically those reasons. And one peculiarity is that it's not just COVID-19. When we had seen H1N1, these are the regions which had reported a disproportionately high mortality, and the cases also were higher from these parts. So this question remains an enigma, and it's a research issue. which needs to be taken up. You mentioned about genetic susceptibility there, but that's a case that requires further investigation. But yes, Maharashtra is it's disproportionately higher compared to the rest of the country.
Yeah, thank you very much for the answer, sir. And one more small question. In one of the slides, we saw that the recovery rate is from 90 to 99% across India, different states. Maybe it's similar to the earlier question. So, what could be the reason? Like so much variation within the country?
No, but the variation, yeah, I look at it from the perspective of because the disease was not uniformly, I mean, in terms of the timelines, it was not the same timeline that all the states were affected. So some of the states which had an earlier peak, they had passed that peak, and the recoveries were higher over there. And if we look at some other states like Kerala at the moment, which is a 90% recovery rate, so because it is daily with reporting around 5,000, 6,000 new cases, so clearly a 14-day period, 17-day period has to pass before these cases would be classified as recovered. So it's the difference in the timelines of the epidemic that has unfolded in different parts of the country.
Oh, thank you very much. That was a wonderful response by you. And the last one more question, with the permission of Dr. Sethi and organizers. One of the participants is asking, like, could you please tell us about the scenario regarding relapse of infection of SARS-CoV-2 in convalescent patients in India?
Exactly. Very important question. That reinfection, not just from, we look at it from the perspective of what are the implications of different strains. How do we know that different strains might have a public health implication? So the only way is our country, more or less, if we are looking at the scenario that we have developed herd immunity. There are various models which suggest that India has already developed that herd immunity level, and therefore we are seeing a decline in the number of cases. So, but what happens if a new strain comes in? Does it have the potential to cause reinfection? For that, we need to establish clearly a definition for reinfection. What is reinfection? So, the current definition of reinfection is unfortunately a bit complicated. We have already proposed that it needs to be simplified so that clinicians and epidemiologists are able to make use of it. So what we are proposing is that we don't go into genome sequencing of the two. So let's say I am infected right now. So the current genome sequencing has to show a different virus, and then subsequently in the other infection, it has to be a different one. Then I can say reinfection. So we are proposing to simplify it, and hopefully we'll have a simpler definition available, and reinfections could be more easily then classified in the community and dealt with it. But definitely we have evidence that there are reinfections in the community.
Thank you very much, sir, for a wonderful response and solving the questions. And over to Dr. Sethi. Thank you.
Thank you very much, Dr. Shantanu. I personally feel that the COVID-19 pandemic has increased the awareness of people towards science. You feel very happy as a researcher when people discuss presently about qPCR, they say RT-PCR, which is, most of the time people are not known to this. They talk about strain of virus and development of vaccine. Interesting. So Dr. Himanshu highlighted the global transmission of COVID-19, including initial report to the present worldwide confirmed cases. He also specifically focused on Indian scenario by providing real data on day-wise growth rate, month-wise active cases, state growth rate, recovery rate, mortality rate, etc. Thank you very much, sir, for providing latest update on the pandemic and India's public health response, as well as future issues and challenges in controlling this transmission. Indian Virological Society and College of Animal Biotechnology will be looking forward for more interaction with you in future, sir. Thank you very much.
Thank you very much, sir, and all the organizers. Thank you.
Thank you very much, sir.
Thank you very much, sir.
So, sir, can we have a break for the lunch time, because
and what is the time?
we have two more speakers.
2:30.
Yes, sir.
Yes,
We
sir.
have two
2:30.
more speakers. Dr. Scott will be speaking at 2:30. Then we have Dr. Panduranga around 3:15.
3:15. Okay.
So we will, break for the lunch, and again we will meet around 2:30.
2:30. Yes. Perfect.
Okay, sir.
Okay. Yeah.
So this link, you will keep it open or you will close down it?
No, sir. I will leave and log off, and again
That's-
around 2:15 I will log in.
Okay. Okay. So we will
Okay.
meet
Yeah.
at 2:30 now, everyone.
Yeah. Yeah, sir. Yeah.
Yes. Take physical lunch now.
Yes.
Okay.
Better we can, sir, let it be on, sir. Or
Leave it as such on.
yeah,
Yes,
we'll,
leave it
we'll
on.
keep as such on, sir. If any.
We will be
Yeah,
back
yeah.
at 2:30.
Yes, sir.
Yes, sir. Yeah, sure. Thank you very much.
Dr. Malik, are you there? As well as he's senior scientist at the Vaccine and Infectious Disease Organization, which we commonly say VIDO. It's an international vaccine center. A protein biochemist by training, Dr. Scott's research interests are in the development of vaccines, especially for protein misfolding diseases such as prion diseases, amyotrophic lateral sclerosis, Alzheimer's disease. He has pioneered a technology that enables high throughput analysis of phosphorylation mediated signal transduction activities in any species. During this COVID-19 pandemic, Dr. Napper served as the interim director of research for VIDO and helped the organization to isolate the virus, develop an animal model, test therapeutics, and ultimately develop a vaccine. The vaccine is named as VIDO COVID-19 vaccine, which is recently approved to begin human clinical trials in Canada. He has also a strong commitment to promoting science interests and education. He is a frequent speaker at high schools within the prominence, as well as four-time winner of the University of Saskatchewan Teaching Excellence Award. Today, Dr. Scott will be speaking on vaccine development in the midst of a pandemic. So, Dr. Scott, again, thank you very much. And I understand it will be, early morning, tough time for you. So but we are very much eager to listen to you. Dr. Scott, please.
Excellent, thank you. Can I get are my slides back up?
You can share screen.
There we go.
We can see
Excellent.
the slide.
So first of all, I would like to sincerely apologize that I wasn't here to give the first talk. That my assistant had put this in my calendar for 9:30 p.m. January 23rd, which for us is actually tomorrow evening. So I had been sitting at home this evening, relaxing with my family, and all of a sudden my email started to go crazy. And one of my classes was writing an exam tonight. So I thought it was just students writing to me complaining. So part of me was thinking, I'll just leave that to tomorrow. But then part of me thought, no, I better check. And then I realized that, yeah, I was missing my seminar. So thank you for accommodating me to come in a bit later. I guess the punishment for me is that it is now just past 3 o'clock in the morning here. So I will do my absolute best to stay awake and relatively quiet, to not wake up my kids, but hopefully we get through this okay. It is an absolute honor and privilege to come back. It was back in 2016, I had the opportunity to visit your institute. And in terms of science, in terms of hospitality, and in terms of food, it really set the benchmark for all of these professional experience. So it's wonderful to be back. What I want to do is to give you a bit of insiders, a personal account of what it was like to be developing a vaccine in the midst of a pandemic. It's one of those things as a scientist, you know, you may think you kind of hope for that type of opportunity. It's certainly been one of the most exciting and challenging periods of my career. And I hope my career is never this exciting or challenging again. I want everybody to think back to New Year's Eve 2019. What were you doing? That for me, we had a quiet New Year's Eve. Me and my family, we went out to a movie. It's always the big debate of what movie we'll go to. Everybody in my family, they like the movies with Dwayne "The Rock" Johnson, especially my wife. So we went out and we saw Jumanji 2. So we were there sitting in this crowded movie theater, not a care in the world. And I think independent of what you were doing, maybe you were at a friend's house, maybe you were with family, maybe you were in a restaurant, in a bar, that New Year's Eve, it's sort of a natural time to think about the year that's coming up, to make your plans, to make your resolutions. And I think it's so ironic that as we were all making our plans, the world had different plans for us. That was the day that we first got the indications that something was about to go horribly wrong. The first indications of these cases of unexplained pneumonia within China. By January 5th, we had a better idea of how serious the problem was. There was nearly 50 people who had been infected, and about a quarter of those people were in very serious condition. By January 9th, we knew that this disease was being caused by a novel coronavirus. Now, this wasn't surprising, but at the same time, it wasn't reassuring that there's a lot of coronaviruses that exist within different species. And for the most part, they don't tend to cause that serious of disease, but they also have a very high propensity to jump across species. And once they're in the new species, that's where you have the real potential for pathology. And if we think of some of the outbreaks that have happened in recent history, so when we think about Middle Eastern respiratory syndrome caused by a coronavirus going from camels into people, when we think of SARS of about 20 years ago, you know, going from bats jumping into people. So certainly, you know, in retrospect, we probably should have seen this one coming better than we did. By January 10th, the genome sequence of SARS-CoV-2 was available. And again, I think we have to remind ourselves just how incredible of an accomplishment this is, that less than 2 weeks after this is first emerging on the radar as being a problem, that we have the most intimate details of the genome of this pathogen. You know, we really can't accomplishment that was. And the reason it's so important is that once you have the genome sequence, you can start to develop a vaccine, whether you're talking about a messenger RNA vaccine or a more traditional subunit vaccine, that once that information is available, that's really all you need to get started. And that's what most of us did. That information was released on a Friday. Most of us, including Ed Vito, we spent the weekend, you know, sort of thinking about, well, what would be a logical way to develop a vaccine? And I remember it so well at that time. There was a lot of debate amongst the scientists of how big of a problem is this going to be? And a lot of people thought, you know, this is just going to burn itself out. This will be gone in a few days. Other people thought, worst case scenario, this might be another SARS. I don't think anybody anticipated just how bad this was going to get. On January 22nd, so just over 3 weeks after the outbreak started, that Vito Intervac was given approval by the Canadian government to work with the virus. And my apologies, this is back when it was still being called the Wuhan coronavirus. Why would a lab like ours, so we are part of the University of Saskatchewan, we're not a government lab, why would we get special permission to work with this type of pathogen? Because we're really, really good at what we do. The Vaccine and Infectious Disease Organization, we have been around for 45 years, dealing with infectious diseases in both humans and animals. Although we're part of the university, we very much have a priority towards product-driven science, that we're not so much interested in the pure research aspect of it. We're interested in doing a science that's going to have real-world implications. And that's best exemplified by, we have 10 commercialized vaccines. We have somewhere near 150 patents, well over 1,000 publications, and absolutely. And, oh, can you still hear me?
Yes.
Yes, okay. Somewhere around 170 people working there, and absolutely state-of-the-art infrastructure. In particular, our level 3 facilities. So a lot of labs have level 3 facilities. We have level 3 facilities that are approximately the size of a football field. We have 10 animal rooms. We can accommodate up to 1,000 pigs under level 3 containment. So in terms of our capacity to work with these types of pathogens, really incredibly unique. We have our own team of veterinarians who does all the animal work for us. And we have literally worked with everything from bats to bison within these facilities. So really remarkable and really quite unique. One of the other things we'll be getting that I'll talk about a little bit later is a vaccine manufacturing facility within our organization. So we're going to be able to go from the earliest points of pure research all the way to generating a vaccine that's ready to go into human beings. So if you want to make a vaccine, what do you need? Well, you need 3 different things. Obviously, you have to have the pathogen, you have to have the virus so you're able to study it, you have to have an effective model of infection. So if you want to be able to test your vaccines or your therapeutics, there has to be an animal model, and you have to have your vaccine candidate. So in terms of the pathogen, we were quite fortunate. We were given clinical samples from one of the very first Canadian cases of COVID-19. And it was a strange, strange day. That, you know, back in the early days of the pandemic, even now, I mean, everybody's making their best efforts to stay as far away from this virus as possible. And we're saying, send it to us, send it to us. But that's what we're here for. I mean, we're there to do those types of things. We have the expertise, we have the infrastructure. But nevertheless, it was an exciting moment, but it was a really solemn moment when those samples arrived, that it's your friends, it's your coworkers who are going to be working with this very dangerous pathogen. And that's certainly something we didn't want to underestimate. We were able to isolate the virus and to sequence it. And we were the first lab in Canada to do that. And that not only enabled our own research efforts, but we were able to share that with other research groups. And to me, that's been one of the things as a scientist that's been most remarkable about this pandemic, is seeing the collaboration and the cooperation between all of the different scientific groups. That this doesn't feel like one sort of group looking for all the glory. This is us as scientists trying to do something to make the world a safer place. The other thing you need is an animal model. Now, typically when we're thinking about animal models, that mice are the cheapest, the easiest to go to, but it has to be compatible with the pathogen. And in particular with these coronaviruses, that knowing the way they get into the cells, that knowing that the ACE2 receptor had to be compatible with the pathogen. And at the time, nobody really knew what the best animal model was going to be. Now, based upon our experience with SARS and with MERS, we suspected that the ferret was going to be an effective model of infection. Now, the challenge is if you want to order mice, you can pick up the phone and you can have hundreds of mice delivered to you, easy as can be. If you want ferrets, that's a little bit more complicated, and that is much, much more expensive. That research ferrets here in North America, they cost about $1,000 each. Now, at the same time, as we saw this, as this was clearly emerging to be a major problem, we knew that other groups all around the world would also be looking at the ferret. And we knew there was a limited number of ferrets that were available for purchase. So we decided we were going to take a bit of a gamble, and we ordered a lot of ferrets. We ordered over 200 ferrets. So we took a $250,000 gamble that the ferret was going to be an effective model of infection for SARS-CoV-2. The other challenge we had, that we bought them from a company in the United States, and they couldn't ship them on the airlines. They had to do it by transport. So they took the back of this truck, they packed it full of ferrets. They had enough food and water for the drive. The driver just drove like crazy to get across the border and to get to our institute. But you can imagine when you open the back of a truck and there's been 200 ferrets in there for 24 hours, just the smell of that was something I'll never forget. And it turns out it was a good gamble, that the ferrets are a very effective model. In particular, you can infect them through intranasal challenge, so mimicking what's going to be happening within human disease. Within ferrets, it causes fairly mild clinical disease. So they don't show a lot of symptoms. They don't show a lot of weight loss. But in terms of being able to quantify replication of the virus within the upper respiratory tract, we certainly see that. For the most part, they're all able to clear the infection on their own. It turns out the hamster is also a pretty effective model. And here you actually see more severe disease. So in terms of weight loss, and you actually start to see some deaths within the hamster. A small percentage of the animals will succumb to infection, which is something that you don't see in the ferrets. And again, not to sound like I'm bragging on Vito, but I'm bragging on Vito that we were the first lab in Canada to establish this animal model of infection. And again, we readily shared all of those results with all of our colleagues. Able to make your vaccine. And we wanted to go with a fairly traditional vaccine. We were looking at making a subunit vaccine. And based upon our experience with SARS and MERS, we had good reason to believe that the spike protein would be an appropriate antigen, that we would be able to get protection. Specifically, it was known that it's the spike protein that has to interact with the ACE2 receptor in order to get uptake into the cells. And that if you can have antibodies that are binding to spike 2, that you can block that interaction. So again, this is something we started as soon as the genome sequence was available, that we cloned the portion corresponding to a fragment within spike, and we started to produce that in-house almost immediately. We wanted to look at 2 different antigen adjuvant systems. So one of these was a commercial adjuvant, and another one was one that we had developed in-house, that based upon sort of similar proteins, glycosylated viral proteins, that we knew could induce protective, long-lasting responses. So off the start, we had sort of these 2 vaccine candidates, that it was the same antigen, but being presented, oops. I'm back, okay. So it was the same antigen, but being presented with 2 different adjuvant systems. So the first experiments, they were being done in mice, and it seemed to be quite effective in terms of the magnitude and the type of immune response we were getting. So we were getting a fairly balanced Th1, Th2 response as compared to other adjuvant systems, in particular alum. And we suspected what we would be needing here would to have a strong antibody response was likely going to be the protective response. So these were certainly encouraging results. And we were also seeing these antibody responses within the lungs of mice. So again, very encouraging that in terms of being able to neutralize the virus, that's what you want to see. And then when we actually went into our animal model of disease into the ferrets, again, very encouraging results that with 2 immunizations spaced apart, we were able to get very strong responses. And indeed, if you compare those to the antibody responses against spike that you see in response to infection, we were getting higher antibody responses with our vaccines. So encouraging results in that we were able to surpass the natural immune response to the pathogen. So if natural infection would bestow immunity, our vaccine should be even better. And beyond just looking at antibodies against spike, we were able to show that these antibodies were neutralizing. So again, within the ferret model. So after 2 immunizations, we were able to induce these neutralizing antibodies. And again, at a level higher than you see in response to infection. So very encouraging results that we were on the right path. In terms of the protection that's afforded, so we looked at it in a couple of different ways. So one was in terms of the ability to inhibit replication of the virus within the upper respiratory tract, looking within the nasal washes. So here you see the results from the 2 different vaccines with the 2 different adjuvants. So in particular, the one shown in red here, that we see a reduction of 1,000-fold in terms of the amount of viral RNA that's present. And for many of those, it took the amount of viral RNA to below detectable limits. So again, this is exactly what we want to see in terms of being able to block viral uptake, viral replication, should not only be protecting the individual, but also it's going to be making them less likely to pass it on to others. It was a bit difficult to measure efficacy in terms of pathology within the ferrets. That again, as I mentioned, there were very few clinical symptoms within the ferrets with no weight loss. So we relied on our hamster model for this. And so what you see with hamsters, unvaccinated hamsters that get infected, you see a fairly dramatic weight loss that's going to be occurring within the week after infection. So they lose about 15% of their body weight. And what we were able to see with our vaccinated animals is that we can dramatically reduce that. And when you looked at these animals, you know, they seemed to sort of shrug off the infection when they had the benefits of vaccination. So again, very encouraging that we were on the right track with this. And again, in terms of first, we were the first lab in Canada to develop and test a COVID-19 vaccine. So that's how we spent much of our year, and you sort of sum it up in a few slides. But it was really a monumental challenge in terms of science and in terms of administration, that you have an organization of 170 people. And even though we're not quite a university facility, we're still sort of burdened by some of the stuff that often slows down university-based decision-making processes. And so here we had to sort of shift everything we were doing. We had to shut down a number of projects. We had to reassign people to be focusing entirely on this project, but it paid off quite beautifully. Quite recently, December 22nd, is that we received approval by Health Canada to start with human trials. And those initiated, it was about a week ago that we started to go into human trials in Eastern Canada. So you kind of put that into a context. I mean, what's the normal timeline for developing a vaccine? Typically, you're looking at the time frame of 10 to 15 years with an investment of, it's usually about $500 million. So the fact that we were able, not even a year after the first outbreak started, that we have this into human beings is certainly something we are incredibly proud of. We are on track that hopefully towards the end of 2021, assuming everything goes well, that this will be a licensed vaccine for human use. So the other thing that was going on while all of that was happening, so VITO is something called one of Canada's major science infrastructures. And what that means, we receive a lot of money from the federal government for things like these level 3 facilities, but with the understanding that's not just for us, that's there to benefit all of Canada and to benefit international partners. So obviously there was a lot of groups around the world who had vaccine candidates, who had therapeutics they wanted to test, but they didn't have the capabilities, they didn't have the facilities to be working with the virus and to be working with these animal infection models. So what we had to do was to really throw open our doors and to help as many of those groups as possible. To date, we've done, I think it's over 70 different trials for outside groups, testing their vaccines, testing their therapeutics. It has been an exhausting year that our veterinarians have been working around the clock. These are very labor-intensive trials to do. Our research technicians with all of the samples they've had to collect. But we couldn't be prouder that we were able to take on all of this outside work to accomplish that for those groups, while at the same time developing our own vaccine. The other thing we were able to do that I am extremely proud of, that back in the early days of the pandemic, there was a lot of concerns that our frontline healthcare workers, the doctors and the nurses, there wasn't going to be enough PPE that was going to be available to them, putting those individuals at risk. So the question became, are there ways that we can decontaminate these masks so they can reuse them to push the supply out a little bit further? And that's certainly one of the things we can do within Intervac, within our level 3 facilities, is we have the opportunity to be able to decontaminate the masks, the face shields. The only challenge of it is that you need all of this equipment separated from each other. You can't just take a pile of it and put in this vaporized hydrogen peroxide. You have to have each mask hanging up individually. So the guy who runs Intervac, he went to the hardware store, he bought all of these lengths of chains, and he set up the apparatus you see right here, so we could go in and we could hang up these individual masks. Effective, but extremely labor intensive. Like, you have to go through and hang up every mask. So we sent out an email to our workers saying, we need people who are willing to volunteer their time to come in, to go through these big bins of sweaty, dirty, potentially contaminated masks and hang them up on these chains. About as unpleasant of a job as you can imagine. Within an hour, we had more volunteers than we could handle. And you can talk about Vito in terms of the number of patents, the number of PhDs, the square footage, but that doesn't tell you what we're about. That doesn't measure the heart of the organization. And to me, this really showed what we're about, that we're willing to step up to do this extra work, not just for our own benefit, but to help everybody out. So a very proud moment. So from the Vito perspective, I mean, what went right? A lot of things. So again, we were the first in Canada to isolate the virus, the first to establish an animal model of infection, the first to develop and test a vaccine, as well as being able to test vaccines for a number of other people, at the same time supporting our healthcare system. What needs to improve? So when you want to go into human beings, when you want to start your phase 1 clinical trials, all of the materials have to be produced under GMP standards. We don't have the capability at the time to do that within Vito. So we were very much reliant on other people to produce the antigen for us that would go into people, in terms of producing the adjuvant we would need. And I mean, I don't know how it was where you are. Pretty much everything that could go wrong in science in terms of delays of just getting the simplest equipment. And so many of these organizations that can produce proteins under GMP conditions, that can give you the adjuvant, they became bottlenecks for us. So as fast as we were able to get through this, we could have been through much, much faster if we weren't reliant on these outside groups to provide those things for us. So that's one of the lessons we learned. We need to be self-sufficient. And one of the things that happened at the start of the outbreak, the federal government stepped in and they gave us the money we need to be able to produce a vaccine manufacturing facility right within Vito. So again, we're going to be able to go from the first stages of basic science all the way to the end stages of producing vaccines that are ready for human clinical trials. That I don't want to say if this happens again, but when this happens again, we will be much better prepared to be able to respond. It's a relatively small facility, but even within that, we would be able to produce up to 40 million doses of vaccine per year. So to be able to make a pretty significant contribution, at least to Canada and hopefully further. Now, obviously, we didn't win the race. We didn't expect to, and I think it's a little bit more complicated than that. I mean, one of the things that's been absolutely incredible to me, working in the area of vaccine development, how it pushes the technology, how it pushes the scientists. So at least within North America, the two vaccines that were first approved for use, the Pfizer and Moderna vaccines, messenger RNA vaccines, that had never been done before for human beings. There had never been a licensed vaccine. But in so many ways, it makes so much more sense that if you can take out that step of having to produce and purify the protein by yourself, if you can let nature do that for you, that once you have that genome sequence of the pathogen available, very quickly, you can translate that into a messenger RNA vaccine. So as you have those types of platforms available, it's really going to increase the rate at which we can respond to these emerging pathogens. The other lead candidates coming forward are the viral vector vaccines. And again, this is relatively new technology that in terms of being licensed for human use, very limited examples, just Ebola, as far as I'm aware of. So the idea that you can take portions of the pathogen genome that correspond to the antigen of interest, put that into these viral vectors and use that as a delivery system. So what we're seeing with the Oxford AstraZeneca vaccines, as well as the Johnson & Johnson. And my understanding is that this is currently being used in India. This is one of the vaccines being applied. So again, this example of how the worst of situations can really accelerate science. So from a global perspective, what went right? This was obviously a time when the world was looking to science, saying, you've been saying all of these incredible things to do. We actually need you now. And I think science responded with a speed and efficiency is something we can all be incredibly proud of. One of the things we were involved with VIDO, the WHO set up these calls where a few times a week they would bring in all of these experts from different fields to talk about the results, to talk about the developments they were making, their strategies, their philosophies. And to me, it was absolutely inspiring to see how people were leaving their egos at the door, that somebody would bring up a problem they have and people would be there with helpful advice. People were sharing results. If somebody needed a reagent, somebody else would chime in and say, it's on its way. So I think in terms of, showing the potential of science, and again, to go from the emergence of a pathogen to having a vaccine produced and going into human beings in less than a year, to me, that's absolutely astounding. And if you would have said that to people a few years ago that could be done, they probably would have thought you were hopelessly naive. So I think that's absolutely incredible. At the same time, hundreds of millions of people are going to be infected. Millions, if not tens of millions of people are going to die. And there's going to be huge economic and social consequences. Can we really call this a victory? I think we need to do better. I think we need an entirely new paradigm of vaccine development. Is this going to happen again? Are we going to have more pandemics in our lifetime? To me, it seems almost inevitable that when you look at the factors that lead to these pandemics in terms of population density, in terms of disruption of environments, in terms of international travel, we're really setting the stage for these things to happen again. So I don't think we can assume that this is going to be the last of these we're going to see in our lifetimes. And in fact, as horrible as this has been, I hope in some ways this is a bit of a warning shot across the bow of how much worse this potentially could have been. It's time for us to do a little bit better in terms of preparing ourselves for this. So how do we get better prepared? So I think things have to happen on two fronts. So in terms of the biomedical sciences, when we think about the current paradigm of vaccine development. It's really based in this idea of specificity. So you want to be able to have these very specific antigens. You want to have those to develop these specific immune responses. Now, the challenge associated with that when you're prioritizing specificity is that you need to know what those antigens are, that you can't start developing a vaccine until that pathogen already exists. And certainly what we've seen in the current situation, that's just not good enough. That even when we looked at the timelines we just discussed, it's not like there was months and months of people sort of scratching their head of should we do something, that there was this immediate response on the part of science, and yet we're left with these consequences. Start developing the vaccine once the pathogen emerges, you've already lost, you're already too late. We have to start developing vaccines before the pathogen even exists. And I think how we can do that, we know that vaccines can induce responses that have greater spectrums in terms of their reactivity and greater spectrums in terms of their protection. And we need to be able to understand the factors that contribute to antibody cross-reactivity and antibody cross-protection. And even now we're seeing that with vaccines to seemingly unrelated pathogens seem to be giving degrees of protection in the context of COVID-19. So we need to be able to understand what those factors are so we can build those into vaccines. Now, it's important to appreciate that there's always the trade-off, that if you want to have a greater range of reactivity, you're probably losing some of the efficiency in terms of neutralizing a virus. But I think this is where we have to change our thinking, that it may be an imperfect vaccine for the long run, but it may be a perfect vaccine that any degree of protection it can afford, that if that can be applied in the earliest stages of the outbreak, that you may be able to contain it long enough to give the efforts to generate those next generation vaccines that are going to be much more specific. So I think we really have to be thinking in those terms, that even what we're seeing now, that these first vaccines that are coming out, it's fantastic they were able to develop them so quickly, and hopefully they are going to be completely effective. But at the very least, they're going to buy us enough time to be able to develop the next generation of vaccines. This pandemic is going to change VIDO forever. That before, so initially when VIDO was first created, it was the Veterinary Infectious Disease Organization, and our priority was infectious diseases of livestock. So, you know, what's of importance to the industry. It was about 15 years ago that we changed the name. So then VIDO stood for Vaccine and Infectious Disease Organization. But for the most part, again, we were a pure research organization. While we commercialized some vaccines, a lot of the work we did wouldn't be much different than you would see in a lot of traditional labs. How we envision ourselves going forward, based upon what we were able to do in this situation, based upon our expertise, based upon our infrastructure, we see ourselves emerging as Canada's pandemic research center. And so a big priority within that is again going to be, how do we embrace this new philosophy of this proactive development of vaccines? And as important as the biomedical sciences are going to be in preparing for the next pandemic, the social sciences are going to be equally important. And I have to fully admit, I mean, I think for all of us over the past 10 years, there's been a lot more push towards these interdisciplinary grants. And quite often it felt when you were bringing in, you know, the economics people, the social science people, it almost felt like a bit of a check the box activity. It was one of the things you had to do. I am now completely convinced that developing an effective vaccine is a critical but imperfect solution, that we need to have all of these other tools in place. We need people to understand and to adopt social behaviors that are going to limit the spread of the pathogen. That when we think of the first 10 months of this pandemic, all we had to, in our toolbox to keep it in check, was people following these rules of hand washing, social distancing, wearing masks. But certainly what we've been seeing is a lot of that breaking down, and we're losing some control of our ability to be able to contain it. What we're also seeing is a lot of vaccine hesitancy. This is a huge concern within Canada. The surveys that have been done, that even once this vaccine is available, there's a lot of people who have enough concerns with its safety, they're reluctant to get it. So it doesn't do you a whole lot of good to develop a vaccine if people aren't willing to use it. So we need the social scientists to help us understand those concerns, to be able to enable us to tailor our messaging so that we can get people on board. So obviously a lot of people were involved in these efforts at VIDO. So these were sort of the core people, the scientists, the technicians, the administrators who have been working around the clock for the past year to make this a reality. And then the people who provided us with financial support. It was absolutely wonderful that both our federal and our provincial governments, they really stepped up for us in a huge way. It was, what do you need? Let's make it happen. I think there's sort of a bit of a recognition that, you know, we always had sort of saying we need to be ready for the next pandemic, but sort of the appreciation now that we lapsed a little bit in terms of the investment that needed to be made. But they certainly stepped up when it counted and provided us with the support we need. With that, I would be happy to take any questions.
Dr. Shantanu, do we have any question?
Yeah, we do have questions. Thank you very much, Dr. Scott. Sorry for troubling you middle of the, it's very early morning in Canada. Okay.
My fault.
Thank you. Thank you very much for your wonderful and a great presentation and a great talk about, vaccine development in Canada. And congratulations. And we appreciate the first institute to develop vaccine. That's the University of Saskatchewan. That's great. Okay. And it's one of the best universities in Canada, I guess. I heard about that. Yeah, Saskatchewan. Two of my friends and senior friends are working in Saskatchewan University, toxicology department. Okay.
And certainly, if there's any trainees who are here, sort of looking where to do their training or their postdoctoral studies, keep us in mind.
That would be great. Thank you very much. Okay, we have plenty of questions, but I will take only a few questions on behalf of participants and organizers. One interesting question, that is, what are the key similarities of the ferrets? You used animal models, ferrets and hamsters. So what are the key similarities for ferrets to human that lead us to work on ferrets to develop a vaccine of SARS-CoV-2 when comparing to the susceptible animals like cats and dogs? Why did you choose ferrets and hamsters as model animals to develop vaccines instead of going for cats and dogs?
Yeah. So part of it, the respiratory system of a ferret is quite similar to a human being. So for respiratory infections, whether it's influenza or coronaviruses, the ferret is a good model on that level. But then on a molecular level, when you actually look at the ACE2 receptors, there's a very high degree of homology between ferrets and human beings. Cats are certainly another very effective model. And in the early days, we were actually looking at doing experiments on cats. Part of the reason we didn't get into that, as difficult as it was to get experimental ferrets, even more difficult to get experimental cats. We also had some concerns. I'm not a vet. When I think of cats, I think of, my fuzzy little tabby who likes to sit on my lap. The vets say that a lot of the experimental cats, one of them described them, he said they're little furry demons, and trying to work with them can be quite difficult. We were even at one point thinking of developing a vaccine specifically for cats. The idea that cats may represent an important reservoir of disease that we may have to deal with at some point. But in terms of picking ferrets, yeah, it was the similarity of the respiratory system as well as the homology of ACE2.
Okay, thank you very much for that. The second question was, sorry for giving you more number of questions. Hope you don't mind taking questions. Yeah, thank you. The second question from one of the students it goes like this: is it possible that the coronavirus that originated in China may lose its virulence in some other countries, and since viruses changes their properties, that vaccine that will be made in some other country work with the same efficiency in different countries? Is it like, can I repeat the question?
No, I think, and it's certainly what we're seeing now is that the virus is evolving, it's changing, it's adapting. And that's not surprising that when you're dealing with an RNA virus, you're going to have these mutations that are going to be occurring. When you think about the number of people who are infected and the number of virus particles that they're creating, I once read a quote, given enough opportunities, even the improbable becomes inevitable. And you're going to get these forms that have these different virulence characteristics. There's also the potential that you could develop forms that the vaccines won't be effective against them. It is one of our biggest concerns right now. So far that hasn't happened, but we're certainly seeing these changes occur within the spike protein. And the idea too, that once you start to vaccinate people, you're going to have less cases, so less chances for mutation, but you're also applying a selective pressure for the vaccine or for the viruses that are resistant to the vaccine. So in a way, you're almost selecting for those. So it would be absolutely fantastic if it evolved in a way to be less virulent, but I think we have to be more concerned it's going to go in the other direction.
Yeah, thank you very much. And the very last question, and a general question, which we are, reading so many social media and all. Some of the companies say this vaccine is 80% efficiency, and some of the vaccines it's 95% efficiency. What exactly this percentage efficiency?
So in terms of blocking the ability for the virus to be able to replicate, and again, you don't need to have 100% efficiency, that these degrees of protection are going to afford protection at the level of the individual, as well as protection at the level of the population. And what we would also expect to see, when I said these viruses are mutating and adapting, that they may become resistant to the vaccine-induced responses, you're not likely going to see, sort of a binary situation where it goes from, it's susceptible to those antibodies to completely resistant. You're going to see decreases in terms of the efficiency associated with it.
Thank you very much. With that, I complete taking questions from participants, and over to Dr. Sethi now. Dr. Sethi.
Thank you very much. Scott, I still remember, visiting Vido, which has amazing facility, state-of-the-art facility, and it is always, it's very fascinating to be at Vido and University of Saskatchewan College of Medicine. Everywhere, I have very good memories. The organizing committee is thankful to Dr. Scott for sharing experiences, starting from the isolation of virus, its sequencing, use of animal model, and identifying the candidate for the development of vaccine. Dr. Scott also highlighted the challenges for the development of next generation vaccines and to remain prepared for next pandemic, as well as the role of social sciences for disease management. Thank you very much for providing very interesting information. Indian Virological Society as well as College of Animal Biotechnology extend sincere thanks to Dr. Scott for accepting our invitation and delivering the talk. And we wish speedy approval of your vaccine for human use. Thank you very much, Scott, and sleep tight.
Thank you so much for this opportunity, and I'm going to go to bed now. So good night, everyone.
Good night.
Yeah, thank you. Yeah, thank you. Thank you.
So our next speaker is Dr. Panduranga Rao, and Dr. Panduranga is Vice President. Dr. Panduranga is Vice President, BioVet Private Limited, Bangalore, and he started his career as a veterinarian in the government of Andhra Pradesh. Joined Shri Venkateshwara Veterinary University, and this was an invitation, and to establish biotechnology labs and master's program in biotechnology. A rare, achievement in his life to get into a university and develop a lab and start a master's program. He has been involved in development of vaccines for both human and animals, and again, this is a very, honor for us being veterinarian that he had been involved in the development of vaccines. closely involved in the development of analytic tests for vaccines and biologics for vaccine and contract research industries. He will be delivering a talk on pandemic vaccine technologies. Dr. Panduranga, please.
Please unmute. Yes.
Was, yeah. Can you hear me?
Yes, sir.
Yeah.
I thank the organizers for inviting me. And my topic, it may overlap a little bit with other people's, this one, but still I will be more talking about in the context of industry, what type of vaccines industry is looking at and how to go ahead. And I will start my PowerPoint.
Yes, you can share. Yes.
Yeah, share. Yeah.
Could you see my?
Yes, sir. Please go ahead.
Okay, thank you. Yeah, I will be talking about these vaccine technologies, which some are old technologies, some actually really evolved during this pandemic, and what all the advantages, disadvantages, how industry is looking at that. Those are the points I will be touching upon. See, coming to these pandemics, this is not the first time. Actually, we are seeing these initial signs much before, probably since 1.5 decades. We have seen SARS spreading a lot initially, but it could be controlled. And then came the chikungunya, probably these temperate countries where these most of the developed countries are there, they are not affected. That's why it is not that much means it didn't get that much of importance, but it caused a lot of disease, and economically also India affected a lot during this chikungunya pandemic. Villages, villages and village, all the most of the people in the village got affected, and it affected their economy a lot. But no, means we didn't react this much the way we reacted for COVID-19. Then came this H1N1. There also, luckily, the mortality rate is very less. It became pandemic, but mortality rate is very less, and we are saved. The world is saved. Then came Zika, again similar to chikungunya. It is limited to tropical countries, so not much attention by most of the this one. And Ebola, again, it started unprecedentedly started spreading to different countries, and most of the West Africa is affected. Luckily, it could be contained. Then came this COVID, means then MERS corona. It is also limited to some particular place, but COVID became a real pandemic. That means there are signs. We are seeing it. It is happening. In the last one and a half decade, we have seen this many diseases with the potential of pandemic or became pandemics. So this same thing we can be expecting in future also. So we need to be careful, and at least COVID is a big lesson for us, and how to handle it and all. Okay, probably some places we mishandled it, and some of the people they handled it very well. We need to learn lessons from this and go ahead. And coming to the vaccine development, the day it's almost a year now. WHO announced that COVID can be transmitted from country to from human to human, and probably by 23rd of last year, 23rd January of last year, it started spreading to the other countries, maybe within the vicinity of China. Okay, so then it took another 15-20 days for that to WHO to announce the pandemic, and probably on 26th of January 2020, we had a discussion in our company in Bharat. Okay, when I say my company, okay, I am involved in Bio. I am a vice president in BioVet, looking after the technical this one. But I am also involved with Bharat Biotech. Both are sister concerns. Okay, earlier I worked there, so that I was involved in most of these discussions and all. So 26th January, we had a discussion. It's on Republic Day. Then we thought, okay, we need to go ahead. Then the question came, what type of vaccine? So then different options we had, and then first thing came to mind is inactivated. But inactivated, that is not the right time, because we don't know about this virus, and the facilities available for this virus are this to handle this virus, and the virus is not yet in India, and no strain is available. But still we kept it as an option. Then there are live attenuated vaccines. We thought that is also of no, not a this one. Then maybe subunit vaccines, because already by 19th January, China announced the sequence. So the first thought will be going to the recombinant vaccines. So we thought, okay, subunit vaccines, vectored vaccines, probably adeno-vector and nucleic acid-based vaccines. Okay, that time we never thought about RNA, but DNA. These three we started immediately. We got the synthetic genes done, and we started these three. And then WHO announced it as a pandemic, and different companies, they started thinking about this. And these different people, they develop different things. And but when we are thinking about a pandemic vaccine, what are the things we need to think about? See, a pandemic today may become seasonal tomorrow. That means what I am suspecting, this COVID may become a seasonal vaccine. So a vaccine that can be used repeatedly, that will be a big advantage in the long run. That means, say, in case of adeno-based vaccines, it may be, or any vectored vaccine, we may not be able to use it again and again. That means as a seasonal vaccine, it may not be useful. So that is one thing we need to keep in mind. If a disease becomes pandemic, probably it will be endemic in everywhere, in every country, and probably we need to vaccinate the people every year. That we need to think about, and that is what is industry also will be thinking. What is this? Whether we will be doing one time or it will be a long term this one. So based upon that, we need to select our vaccine candidate or vaccine technology. So then the other thing is how long it is going to take to develop. So in vaccine development, we have a few stages. Probably with all this one, shortest time is probably to develop the candidate vaccine up to preclinical. The clinical trials are going to take long time, and that is what happens most of the times. And then next is whether we can produce this vaccine, whatever the technology we do. Say in case of RNA vaccines, see, we do not have the technology, so that is out of question for us that time. So whether the scalability is another question, and suitability of our facilities, whatever the industry has already. So we thought, okay, any vaccine that is subunit, which can be used in baculoviruses, so that we can produce baculo cell cultures, we are very much aware of, or the lot of experience is there. So cell culture based vaccines, that's what the main thought we started with. So what all those things, okay, we thought, okay, probably we can go with, because sequence is available, probably we can go with baculovirus thing. And then we thought, okay, ad5 based technology already we were using for something else, and our lab is very much involved in this ad5 based this one. And so we thought, okay, ad5 technology we will see, but still we are having our own doubts, because ad5, when we tested within our people, within our campus, how many, all the people and all, almost 25% people are having antibodies against ad5. So we are little bit skeptical about ad5, and we thought, okay, we will think about other things, especially baculovirus and all, because those are the technologies that are suitable for us to use already available infrastructure. So then the safety. especially by the time by April, the virus is there in India and it is isolated, and different institutions started coming to us thinking, okay, whether we can do something with this isolated virus. Then immediately we thought, okay, the major issue with is the infrastructure available. Okay, we thought, okay, let us think about inactivated vaccines. Okay, with this, whether we have the infrastructure and safety, these are the major issues. So then in the meantime, we collaborated with different universities worldwide about using vector, something like using influenza virus or using rabies virus as a vector, using Newcastle disease as a vector. All these things, these proposals came, and we kept ourselves open, because all can be developed, all can be means developed using the available infrastructure. To scale it up, to produce in large scale, the available infrastructure is there, so that we started thinking, we kept our this one open. So anyhow, during that time, already this is most of the people might have discussed about this, and the spike protein is the target for most of the things, and immediately we got the spike protein synthetic genes. We put it into E. coli because that time there is no antibody available. There is no reagents available. By the time we get some technology, we need some reagents to identify whether I have the virus or antibodies are there, or all these things. So our initial thinking is to develop the reagents. Just we put the genes into baculovirus, we put the genes into E. coli, injected some animals. That is the first thing we did, and boosted it and waiting for the antibodies. By the time all these things happen, probably it is April, and then we are looking at different technologies that by the time most of these platforms, most of the manufacturers announce their technologies and all. And if you see this, most of the people, this is maybe by April, what I am talking about, April, May, most of this is from WHO, most of the things, most of the manufacturers announced are VLPs. Probably it is faster. The sequence is known, so that everybody jumped into subunit vaccines, VLPs. And surprisingly, RNA also, there are almost 20 companies, more than 20 companies, they started, they announced RNA vaccines. And the next came the adeno and DNA. See, all these first four, they are all based upon the recombinant DNA technology. And why they announced that many is because already sequence is available. So to quickly start any vaccine, probably these are the first things to be selected. But there are, next thing is that inactivated vaccines. Inactivated vaccines, very few companies they announced, and most probably because it requires a BSL-3 facility, and most of the manufacturers are not having that BSL-3 facility. Or luckily, these veterinary people, most of these foot and mouth disease vaccines are produced in BSL-3 facilities. And for Bharat Biotech, it is luckily we wanted to produce polio vaccine, inactivated polio vaccine in BSL-3 facility, and the facility is getting ready by that time. By February, March, we have the facility ready for polio. But by the time we have the thinking about other things came, means other ICMR institutes and all, they came up, and we started thinking about inactivated also. But other than that, except for inactivated vaccines, probably BSL-2 facilities are enough, or that is, and normal GLP facilities are enough. So probably that is the reason most of the people, they entered into nucleic acid based or VLPs, RNA, adenovirus, DNA, and other things. So this is this one about, sorry. These are the Indian manufacturers, they announced their vaccines, and I will tell you what are the stages and all. This will give you some idea, or it is true with international, this one also. It will give us some idea how, which vaccines are won in the race. Okay, by the within 10 months, most of the means we got some vaccines out. That means they could race at the time required for to develop those vaccines are definitely winning this one. And see, we started, Bharat Biotech started inactivated vaccines, and it is in phase 3 clinical trials, and it is approved for the vaccine, means approved for to give it to as a, to give to the people as an emergency vaccine. Okay, and Serum Institute and Indian Immunologicals, they announced codon de-optimized attenuated vaccines, but it is still under, I believe, in preclinical trials only. They didn't enter into the clinical, so it is not that easy. This virus is quite big, and reverse genetic systems for this virus is, it is not that easy, but still the safety is definitely a concern. Whether really in attenuation is happened, to check the attenuation itself is a big task. So I believe because of the time frame, this one didn't go much further. And there are different viral vector vaccines. They are replicative defective vaccines like Chimpardino, which Oxford University developed, and in India, Serum Institute has taken it up, and okay, it is. This is a definitely front runner, one of the front, this one, and it is already, it is in the market. And this is, it didn't happen simply like that. There is lot of work done before this pandemic. Already the vaccine, it is used to develop vaccines for other diseases, and the technology is available, the know-how and the a lot of experience is there available with this. So this became a front runner definitely. And Bharat Biotech, we also started AD5, but it never went beyond preclinical, because already we know that most of the people are having pre-existing antibodies. And we also started influenza, that also didn't go very well. Then that Reddy started this Russian vaccines, they started here, they brought it here. It is also adeno-based vaccine, and it is now in clinical trials. And RO vaccines, they are Aurobindo Pharma. They started about vesiculovirus vaccines, vesiculovirus vector and all. That is also now in preclinical only. Then Zydus, they announced measles, that also didn't go anywhere. And Bharat Biotech announced rabies based, that also didn't go anywhere. And biologically, they put that S protein in Pichia. I think they got permission for the clinical, I'm not sure. And Mynavax, somebody, they the RNA and DNA vaccines are also announced. And Zydus Cadila is one of the first one to announce the DNA vaccines. That is in clinical 2 stage. But at present, what we can say, in India, 2 vaccines are there which are now approved for vaccination, manufacturing and marketing, though they are for the emergency use. That is, one is inactivated vaccine by Bharat Biotech, and the other one is Chimpardino vaccine, AstraZeneca vaccine of Serum Institute. Both are there now in the phase 1. They are doing the vaccination for frontline workers, and it is great. This is the achievement is it's not a small one. It's hardly 10 months they could do. We could bring a vaccine from the scrap to, we could bring a vaccine to the market. So the inactivated, okay, I will be going through different vaccines that we used. And initially the inactivated vaccines, the technology is known, the facilities are available, but for SARS-CoV, COVID-2, the most of the people are not having this facility, BSL-3 facility. But people who are producing polio vaccines, they be able to do it. So That is one advantage, and the big disadvantage is biosafety and handling. So the initially our people used to wear those suits, complete suits with uh, air circulation and other things, and it is very difficult to work with those suits for 8 to 10 hours in a lab. And when there is no vaccine and we don't know much about the virus, how pathogenic it is and other things. And they are the heroes. They are the heroes who did it and who went into the BSL-3 facility with all these suits and produced this vaccine. And the other one, codon optimizer, already discussed about it, and a little bit I will go into this. This is a good system. As we know, amino acids are coded by more than one codon. That is what we call degeneracy. And some codons are more preferred than the other codons. So this is one technology people started thinking to produce attenuated vaccine. So if I can produce an attenuated vaccine, it is a great this one. If it is safe, you can use it in probably, you need not go for a injection. You can go orally, intranasally, or some other way. So vaccination will be very easy, and the production also. Live vaccines production will be little bit easier, or you can produce more doses per given facility available. So that is one advantage. But the biggest disadvantage with this one is how you are going to show that it is really attenuated. That is the biggest disadvantage. And in case of SARS, this one where the model animals are not very well defined initially, or even still today, this is an issue. And Chimpadino, yes, it is a front runner in the old technologies, one of the front runner. And biggest advantage is means the moment they come to know about the sequence, they could put into that one, and the vaccine development, the virus development took very short time, probably 2-3 months, not more than that. Okay, then started these clinical trials and other things. They have selected Chimpadino because if you are selecting a human adenovirus, it will be having, people will be having a lot of pre-existing immunity. So they selected chimpanzee adenovirus. And yes, this can be used as a pandemic vaccine, but one-time use. Okay, suppose if you want to use it for a seasonal vaccine, it is not a suitable candidate. So that is one of the reason we also skeptical about these vectored vaccines. And when things moved a bit better, when the strain became available, we moved with the inactivated vaccine because that can be used as a seasonal vaccine if required tomorrow later. It can be used for years together. And whereas Chimpadino, you give for one time, probably for a pandemic, it is good to use it as a once in lifetime. Okay, suppose if we get another pandemic, it may not be the same Chimpadino serotype we may not be able to use. Maybe we need to think about something else. So these are the little bit of disadvantages of these vectored vaccines, where pre-existing immunity will become a big issue. And especially in the pandemics, if you are vaccinating all the population or most of the population, this can never be used again, or the same serotype can never be used again. And I'm not going much into this one. Chimpadino, this one is a replication defective, so that it can infect the cells where the virus is present, but not the human cells, not regular human. This one, it can enter, but the virus cannot be released. That's what is the replication incompetent. And yes, this is a good candidate for at present as a vaccine candidate, but adenoviruses, you need a lot of this one antigen we need to push into the body, and adverse reactions are also there. But we need not worry much about these adverse reactions. They are not serious adverse reactions, not many. only disadvantage of this particular thing is you cannot use it for as a seasonal or in later situations. And this is another vaccine, VSV based, people used it is a replication competent, and it is used for Ebola. Though we have an experience with Ebola and other things, somehow it didn't happen the way, the faster way, the way it happened for adeno or other vectors. I don't know what is the reason, but definitely it is a good candidate, but one-time vaccine. And this is one technology that came now. We miss 2020. It miss the people were thinking about this one, and none of the up to now there is no vaccine available based upon the RNA. This one before 2020. So people started thinking about in 2020. Probably I will say it is born in 2020 and conquered whole world. Okay, and this is what happens in pandemics when you are if it is not this pandemic, RNA vaccine should would have been taken another 10 years, a decade, to come into the picture. Just because of this pandemic, these RNA vaccines, they are several different people, they started these, and at least 2 vaccines are in the market now. And the fastest way, these are the probably is the very fast developed, very rapidly developed, and they are in the market. And it's a great story actually, these RNA vaccines, because within 1 year a new technology, they started it, they scaled it up, and now it is in the market. It is being used by in different countries. And probably this is one technology that is going to change the whole vaccine field, I believe. But still it need to be evolved, especially for scale up and other things. And definitely it is a big this one for future. And DNA vaccines, this I am hearing since maybe 30, 40 years now, maybe first time in 90s, maybe 30 years. And probably what I think is with this pandemic, COVID-19, these were one of the technology that is going to die, I believe, but I'm not sure. But what that is my personal opinion. And it couldn't deliver anything. No vaccine came up to the market. And this is the probably the first manufacturers announced most of the vaccines that are announced are DNA based vaccines. That means there are several people, they started with DNA vaccines, almost 30 to 40 manufacturers, they announced about this DNA vaccine thing. If it is not coming for this, if it is not working for this pandemic, or if they cannot bring it out, probably this is the last this one for this DNA vaccine. That's what I am thinking. And probably emergence of RNA vaccines is a big blow for these DNA vaccines. And generally, when we see, when as a manufacturer, when I see the vaccines, generally it takes 20 years, 15 years, 20 years. That's what people think. And if this is the regular vaccine for any this one, we could have seen this thinking in 2020 about a technology. It could have come out to the market in 2036. That is 15, 16 years. But less than 15, 16 months, these vaccines, these pandemic vaccines came into the market. That is the greatness and the way people worked. And this is true with any other vaccines. COVID initial goal was 18 months, and actually it is surpassed. It is only less than 12 months. Less than 12 months, the vaccine is in the market. Probably this is the fastest vaccines that became available for the people. In spite of means, it is not that vaccines are pushed into the market without testing that safety. All safety tests are, the trials are done as per the requirement. Safety is not compromised any place, but only thing that is done is the regulators. The major issue of this vaccine development is regulators. you submit your clinical means you submit your documents to the thing, and regulators take longer time to approve. Between phase 1 and phase 2, it takes approximately 6 months for the regulators to give the approvals. That's what happens in a normal situation. Whereas here regulators, especially the Indian regulators also, they work day and night, and they 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 went through all the documents. The committees went through all the documents, and it happened. They took least amount of time actually. That is why the vaccines the vaccines are in the market, and this is what happened. Uh, this okay. For what is WHO product profile that is required and all, actually they thought. Uh, this is active immunization that is already known, no contraindication. See, this is preferred, whereas this is critical, that is acceptable. That's where is the, what we see the safety issues. See, in case of regular vaccine, a seasonal vaccine, suppose something like what we are seeing with other vaccines where we need to use analytics before taking the vaccine. That type of this one is not allowed generally for a regular vaccine, for a seasonal vaccine. Because it is pandemic, some amount of adverse reactions are acceptable. So, and there are other issues, sorry. And route of administration generally in, say, non-parenteral is nasal or oral is preferred, but IM is accepted. Most of the vaccines are now IM, and Bharat Biotech is now coming up with a nasal vaccine. Let us see how it goes. And product stability, okay, if I can give a product at room temperature, that is the best one, but inactivated vaccines can be stored at 2 to 8, that is acceptable. But some of these vaccines, RNA vaccines and all, you need freezing temperatures, liquid nitrogens or dry ice and all. They are a little bit difficult. Probably RNA vaccines, I believe that will go up further. And this is one area in the RNA vaccine they need to think about, how to make it stable at room temperature or 4 degrees or something like that. And multi-dose vials are given, and the important thing is scale up things could be done. And I am again coming back to my second slide, where how these technologies. So coming to the manufacturing facilities, so how have it happened is chimpadinovirus. So all you need is manufacturing technologies like fermenters to produce in large scale, microcarrier systems or the suspension cultures. Luckily, these 293 cells can be produced in suspensions. In large scale fermenters, it can be produced. That's the reason that could be scaled up. So that is one of the important positive point in case of chimpadino or any adenovirus vectors. So they could scale it up easily. Whereas in case of killed vaccines, the scale up is became a little bit problematic for us, but finally we went with microcarriers and in the fermenters. Initially it was done in the flasks, later it went to microcarriers and fermenters. That is what is important. We should be able to use the facilities that are already available. Otherwise, to create a facility, to create a GMP manufacturing facility, that itself will take 2 to 3 years. So any technology that can be produced in the existing manufacturing facility will be a big advantage. But in case of RNA vaccines, there are no manufacturing facilities, but the facilities required are minimum. You are not using any fermenter, the other things, ready to use systems became available. So that is the success. That because of that, RNA vaccines became a big success. And then the large scale production, already I discussed, and large scale vaccination. This is also important. To do large scale vaccination, you need, how are we going to create the infrastructure to transport the vaccine, store the vaccine and vaccinate? So for all these things, that is one disadvantage what we see. If we are going for RNA vaccines in India, probably our infrastructure to store it will be a big problem. So maybe we need to develop further infrastructure for that. But whatever the vaccines we produced in India now, the inactivated vaccine of Bharat Biotech or the adeno-vector, they are at 2 to 8 degrees and the all infrastructure is available because of our vaccination programs already in place. So as already I discussed, different systems are being tested, different technologies are tested during this time. Almost 150 manufacturers or 200 manufacturers, they announced it and they tried their best, and very few of them became really means they could bring the vaccine into the market. And now it is very difficult to do even clinical trial. There are no volunteers available to go for clinical trial. So time is important. How fast you can bring the vaccine into the market is important, or to the clinical trial level. That is what is most important in the when we are selecting a vaccine candidate. Now I am hearing from different this one, 1 million people are required for the clinical trials all over the world to test all the vaccines which are at that stage, but people are not available because already some vaccines are in the market, and people will prefer going to get a vaccine which is proven, then go for a clinical trial. So these are the this one we need to think before starting any vaccine program. Yeah, this I think already discussed, and thank you.
Thank you very much, Dr. Panduranga.
Do we have any questions?
Yes, sir, we have.
Dr. Ratan, go ahead, please.
So, Dr. Ram, thank you. And I have two questions from the participants. One is from Pinak Lamania. He's asking which authority give permission of clinical trial, and what is the difference between clinical trial 1 and 2?
Can somebody repeat that one? I am unable to hear.
Sir, he's interested to know what is the difference between clinical trial 1 and trial 2, and who is the competent authority to give the clearance for these trials?
Yeah.